We get experimental science. We're curious, non-judgmental.
We're going to have Danny on in just a moment, but we are going to be chatting about a lot of different papers. Hello, Pinky Giblets. Welcome in. I'm really looking forward to, we did our first crossover about 2 weeks ago, and it's a lot of fun to chat science, especially both of us are learning about each other's fields and the unique approaches that we have on hand. Also, I might just say, both of us having our unique frustrations with the other's field about why not doing this, why not doing this other thing, and then limitations of technology. And so I think it's been really eye-opening to be able to do this kind of crossover, and big thank you to Danny for doing this, because I think it's an absolute blast to be able to talk science. So just to give y'all a little preview, we'll be talking about Tyrannosaurus lineages as one of the news stories. We're also going to do troodontids. Hugen, we might do that on Monday for looking under the microscope. We'll definitely show them next time, because I want to make sure we look under the scope for that, Hugen. I don't want to sell you short on that, Hugen. Indeed, Gojira.
The Tradoon.
We also got two papers, one on aging in insects, so older parents producing distinct and unique offspring in two different species, and it's a way to study the aging effect in us humans as well in a different organisms. It's a model genetic system, as well as looking at fruit fly and mosquito fertility as a function of a Wolbachia infection. And the Wolbachia is an endosymbiote, and it's really, really weird and just give y'all a quick look to it. Be excited about is if you perform a mutation on these animals that makes them less fecund, so far fewer eggs being laid. It turns out, I'm going to invite Danny. It turns out that these animals, once you infect them with this Wolbachia, they're able to lay better again. So it restores. So it's a host manipulation, essentially, is what ends up happening. The host is manipulated to lay better and produce better embryos. So those are some of the topics that we'll be chatting about tonight. Please have your questions ready. Quiz us, teach us, ask us questions. That's why we're here. And again, big thank you to Danny for doing this with us tonight. I'm so excited. Been looking forward to this all week now. And actually, yeah, because last week we didn't do it because of Thanksgiving. And I was like, oh, curses Thanksgiving. But no, I love Thanksgiving. I did deal with a bout of food poisoning, but better now. A fountain of youth? That is a great question, Ubi. They did not look at aging in this particular study. But that would have been it. Actually, Ubi, the other thing, too, that they don't mention is that Wolbachia can cause a lot of bad things as well. Hi, Sparky Pugwash. Welcome in, Sparky. All right, there he is. How's it going, Danny?
It's going well, Blinn. How are you doing?
I'm all right. Let me make you much more giant, sir.
All right. Embiggened.
There we go. Thank you so much
excellent.
for coming on. And I've been really looking forward to this.
Me too. Yeah. I'm beginning to wish I'd sent you a better second paper, but it'll be interesting.
I think both the papers that we're looking at tonight have unique features that make them exciting to look for very different reasons.
Yeah, I agree.
And I think the T-Rex paper has a lot of implications in other fields as well.
Yeah, I don't necessarily agree with the conclusions or the methodology of that paper, but I think it's going to be some good discussion.
Yeah. So everyone, welcome in. If y'all don't know, Danny is the paleontologist extraordinaire on the internet. Stephen, the shark, thank you for the tier 1 gives up to Movember. Thank you, Stephen the shark. Danny, single greatest science communicator on Twitch, on YouTube, on the internet. He's here. He can't deny it. I'm allowed to say this. And it's an honor and a privilege. You make me blush.
My goodness, Blinn.
It is a privilege to have you here, my friend. Thank you so very much.
It's a privilege to be here. Yeah. Tremendous respect for everything that you do, Bullet. Obviously, I've said it a million times. I'll say it again.
Right back at you, my friend. So what's really cool about this, y'all, is we've both selected 2 papers from our fields, sent it to one another for us to read over, and, assuming we don't have the expertise always on these studies, but we're going to chat about them and look at them from the perspective of our own fields. And last time this was a lot of fun because we analyzed, genetic studies from the paleontological perspective, paleontology from the genetics perspective, and I think we both gained appreciation for not just the science, but the tools that we have available, and also had tool envy a little bit on some of this stuff.
Definitely. Yeah. It's really fun to kind of explore what common ground we have, even in very disparate fields, and then just like the surprising differences that there are between our fields also.
And maybe what common ground we should have, but don't currently.
I agree. Yeah, because genetics doesn't have a great deal of overlap yet with not with dinosaur paleontology, because we don't have any dinosaur genes yet. We don't have any DNA from any Mesozoic dinosaurs, but some of the methodologies and stuff, there's definitely overlap. And it's funny because like paleontology has direct overlap with so many other scientific fields, from physics to chemistry to lots of other scientific fields, but not with genetics yet.
So one thing that in the genetics, just right off the bat, that isn't as clean is the morphological studies, right? So like the way, like we were talking last week about all the measurements being done to really identify if this is a true dinosaur or not, of a new species, and like the categorization that you undergo. Sure. There are plenty of studies in genetics where it's like, this is a new species, and then how much morphology have you done?
This is a dinosaur.
None.
This birdie.
Right.
Birdie dinosaur.
It would be great to have
dinosaur
that.
birdie.
You don't even have to do
birdie
that.
Birdie dinosaur. Dinosaur birdie birdie.
Yeah.
It would be great to do the extra, I think, metric and measurement to have that level of data, but it sometimes just isn't done because it's almost taken for granted, like it's a lost art.
Because you've got the genotype, like why do you even have to worry about the phenotype?
Yeah. Which
I
think
phenotypes are important. Like it's important to understand that stuff too.
Yeah. So that's one of the reasons I picked one of these papers tonight. It has a little bit more like pretty pictures and focuses a lot on the more on the phenotype and not just the genome. But you tell me, where are we starting tonight, Danny?
Should we start with one of yours?
Sure. We want maybe we'll start with the aging one.
Okay, yeah.
I think this one...
A lot of this sailed over my head, so it's gonna be cool to ask you about it.
It's
yeah.
So both the papers are brand new. They came out in the past 2 weeks. And I had a little background for this. One of my best friends, when I was an undergrad, he was a postdoc, tested this hypothesis on accident.
Huh.
So he had fruit flies that were in vials, where you kept them in the lab because you keep moving them to the same vial, new vial with new food over and over again to propagate them, and it's the same genetic line. So everyone in that vial is genetically identical. They're inbreeding like crazy, which I thought as an undergrad, I was like, how are they not just like legs coming out of their faces?
How are they still? Yeah.
And it turns out insects are very resilient to that.
Interesting.
And so you can just inbreed them. You can have everyone genetically identical, and it's a really powerful tool.
Are there just a lot of like copying errors when they're producing zygotes, or is it?
So it's actually the error rate is lower, it seems.
Oh.
And so that's why you can just keep inbreeding them. And honestly, even behavioral issues have not really arisen. It seems like with other organisms, like mice in particular, are notorious for inbreeding issues. There was famous study, they're trying to look at learning and memory in mice, and it's associative memory, right? So you pair an odor with a sound, or an electric shock with a sound, or what have you, and they were like, oh, we found this mutant in this mouse, like this mutation that is learning deficiency, and how cool is this? Turned out the mouse was deaf because of And it was just a mutation it had because of the inbreeding. And it's
Uh-huh.
like, well, there goes the conclusion.
Jeez. Yeah.
There is deafness in some fruit flies, but it's interesting that we have the resolution in terms of sequencing of knowing if it's wild type or let's say normal in terms of the genome. Like there's nothing that's causing some kind of phenotype that's negative, which is difficult because like, well, are you looking for it? And then there's, you can break any gene in the genome, and then there's mutants available for that. And so you actually, it's backwards for the genetic naming. So the gene for the mutation that gets rid of pigment in your eye cells that makes them white-eyed, that instead of the gene being called red, because that's what the normal function looks like, it's called the white gene. So it's whatever the mutant phenotype is what you name them. And so like Sparky's asking, well, how does one test for deafness? So Sparky, it's our famous fly called yellow white because it has a yellow body and white eyes as its mutant phenotype, and when the males and females mate, there's a courtship song, and the males aren't as good at doing the singing because they can't hear themselves versus other.
Really?
Yeah. And so even like that level of resolution you can get down.
That's pretty cool, actually. Wow.
Yeah, and so it's again like the tools that you have access to, but it's almost imagination. Octavius, thank you for the gift sub to Danny is the bomb. Danny is the bomb. I don't know which Danny got the gift sub, but our Danny is the bomb. Thank you, Octavius, and hi, Corn Relish. So, when my friend he had this theory that older flies, when laying embryos, their babies would be different than younger flies.
Okay.
And so the idea is similar in humans. And actually, this paper even cited that there are human studies demonstrating that if you're older than 50, the survival rate of offspring is lower in humans. There's also increased risk of cancer, diabetes, and Alzheimer's if you're over 50.
In the resulting young?
Yeah, in the offspring.
Is that to do with the gametes just being older themselves? Like I know I wonder about in flies if it's like in humans where you're, after you develop those eggs, you've got basically the same eggs for your whole life and you're just kind of slowly churning them out. Is it the same with Drosophila?
So it is not. So actually we can I like the two papers because they kind of went hand in hand. I can show you all images of a fruit fly ovary. So this is a fly ovary. Ignore the colors right now. It doesn't matter. It's a pretty picture.
Okay.
It looks like a conveyor belt, right? Like you've got little cells and then as beads on a string that keep getting bigger and bigger and bigger. And so these really tiny ones at the tips are called they're the embryonic stem cells, and they divide asymmetrically. So when they divide, one stays a stem cell and the other one becomes an egg. And so flies and most insects don't have a set number of eggs. They keep producing them constantly. And so it's not like in humans where you have a buildup of... Hi, Irish John. John likes to cause trouble, so it's not like it's a constant buildup, like a one-time buildup of the egg number, but rather you're constantly producing them. Now, there are effects of the ovaries.
Hello there.
Like on aged ones.
Hello there.
Jaded. Like aged ovaries are different in flies. Like there are some chromosomal abnormalities, but in the flies it's much less because they've been stored for too long. It just seems like that original division and the growth, because there's a lot
the
of
ovaries are just, they get less good over time at producing high quality eggs or something like that.
Exactly.
Okay.
Yeah, so it's a little bit different from how the humans work, but it's still having that aged animal and the effect on the offspring, which I think is really cool.
Right.
And so
interesting.
My friend had decided to test if the embryos, the offspring, of these aged parents had a different immune response. Was it not as good of immune response? And so he had actually found that, these parents had like really, really like good immune responses, but the babies were terrible if they were from old parents. And he was like, that's so cool. So he gave the flies to his wife, who was in a different lab. He's like, hey, could you retest it with your flies as well? To make sure it's an effect. And she retested it, and it didn't happen at all.
Interesting.
And it turned out it was because the rate at which they were putting them onto new food was different.
That was enough to cause a different outcome entirely?
Yeah, so this is one of the reasons why I was interested in this paper, is because actually in the methods section, they don't talk about how often they were put onto new food while they were being aged.
Interesting.
It just said as needed. And so I'm like, and this is one of those events that it's not published in Science, right? Because it's a negative piece of result. No one's publishing it. And so it's like, I wonder what the result would be. And Sparky asked if it matters old male compared to an old female. So Sparky, for this, they're aging both the male and female flies at the same time. And so they have 20 individuals in a tube. And Danny, I was going to ask you this too from chat. What do you consider an old fly to be? An old fruit fly?
I don't know what their lifespan is. I guess I don't know. Would it be several months or something would be old?
So I've been asking folks, I've heard like the whole gambit. Like Dan here says 2 days and months. In the wild, it's about 2 weeks. But that's because there's so much other stuff that's killing them, right? There's weather changes,
high mortality rate from predation and
dehydration, and just like whatever you can come up with. In the lab, it's 3 months, about 3 months. So you're right on point. What I would call an old fly. We did experiments in grad school. It was 90 days old, and that was our old fly. So like, you're aging them for 3 months to get a result, which is, by the way, in mammalian studies, they would kill for the opportunity to do 3 months of aging.
Oh yeah,
because they do years. But here it's 20 days. Is all they did.
Not very long. Wow.
So it's I'll show you all the graph here. They actually did a really nice outline of when exactly they're doing the various experimental setup, and so of where they're grabbing the eggs, like either at day 3 of the lifespan of the individuals or like day 21, and then they keep repeating that over and over, and they take offspring from those pools. One batch they use to do the next experiment, the next egg lay. The other batch they use to test, lifespan and movement and just like quality of the offspring, essentially. And so I thought that was quite a clever way of doing that testing, right? Because then you have 2 pools. You can have them continue laying, but then the other one is you're actually doing like testing on these organisms and seeing like how the aging process is affecting them or not.
You've got a really nice control there.
Exactly, yeah, and they did the short generation time as well as long generation time. So they asked both. And again, what's I think extra cool about this study that I really like nowadays in genetics, and I wanted to ask you, Danny, if they do this in paleontology as well, is the use of multiple organisms to validate your hypothesis. And so I guess in paleontology, what I would wonder is when you're describing a new species, like how much maybe cross demonstration of other uses of organisms are. Like here's the Tyrannosaurid. Are we going to compare something that we think is closely related and try to prove like they are distant, or is it just like how often does that multi animal come in?
So you're not talking about like how many individuals of the same proposed new species do you need to have? Are you talking about that, or are you talking about like looking at close relatives to compare?
Yeah, like close relatives to kind of further the hypothesis that it is different.
Oh yeah, we definitely do that a lot. That's kind of a requirement. Is that you compare it to other close relatives, and then if you're going to claim that it's a new genus or a new species, you've got to name a bunch of what we call autapomorphies, and those are just like unique characteristics, where it's like this dinosaur is new and unique. Here's the traits that are new and unique about it, and so different authors will have different numbers of autapomorphies that they're comfortable with. Some, you know, for some people who are really eager to name a new species based on like a single tail vertebra or something, they'll be like, "Oh, well, we found one autapomorphy, so it's definitely a new genus," you know. And it, yeah, yeah. Does that make sense?
Yeah. So it's your argument is stronger if you have these more examples between other species.
For sure.
Yeah.
Yeah. So if you can compare it to other members of the same family, for instance, and say, well, they all look like this, and this one's a little bit different. Yeah. But sometimes it's really difficult to find overlapping material because, you know, you might not have the same part on these different dinosaurs. And so you're kind of left in the dark there.
So I guess that's similar to kind of to this study. And hi, Dan. Welcome in, Dan. That they were looking for another organism that did the same behavior or the same phenotypic outcome when like a similar experimental paradigm. So it's not so much proving that these are two different organisms, but rather that what we're observing isn't unique to one species. And so like having that multiple species testing, you know,
you can say that that's a real phenomenon. You could be confident in that assertion if you can show it in different groups of organisms.
Or you know, a reviewer can come back, chat, because we have, you know, these are peer-reviewed papers, and say, well, is this only in bugs? Why do I care? But if you demonstrate it in multiple species, it's probably more likely.
That's really exciting. Yeah.
Yeah, and it's more likely to be conserved maybe in like, and also like I mean these two species they tested are cheap, right? There's nematodes, which are you put them on an agar plate. And if you forget about them for 6 months and rehydrate them, they come back to life. That's their like those are
very cool,
inherently like cheap and amazing animals. And then flies,
pretty resilient,
like 3 cents a day to keep alive on average, right? So versus like the mice, it's like $75 a day, like for a single mouse. You know, it's. But with nematodes, I thought this might be a cool biological phenomena as well. Every nematode studied in the world of c-elegans originated from one lab in the UK, from a gentleman called Sidney Brenner, who was like, I think this is cool. This one worm that is hermaphroditic. So you know everyone is genetically identical because it's self-fertilizing.
They're all descendants of that one individual nematode in that laboratory.
Yep.
That's
How cool
pretty
is
wild.
that?
Wow.
And what's really nice about that is when you say wild type in a worm, everyone knows what you're talking about. Because there is only one wild type, and every mutant that they have available in these worms is made in this one worm.
Interesting. Wow.
And so they're, you know, they have sequence.
It's nice to have such a robust control there, where you can say that these are all genetically identical to this one original worm. That's pretty wild.
Yeah. And so, you know, there's still this phenomena of effects of how you keep them. Presumably there's going to be, like, how you treat them in your lab, there might be some genetic drift. You know, like inadvertent selection if your incubator running's a little hot and then there's a couple mutants that arise that do a little better. There's going to be some variance, but it's nothing like...
Right.
It's not presumably a huge variance in this. And which is unlike fruit flies. There, when you say wild type, I can list off 10 different wild types that are in papers. And like same with mutants. They're made in all kinds of different genetic backgrounds.
And that's because the original, like, fruit flies are not hermaphroditic. They reproduce sexually, right?
Yep.
So you have to have that shuffling of genes in order to make new fruit flies.
So you can
kind of lose that control aspect.
Yeah. So that it's the term for I'm sure they use something different in paleontology, but it's isogenic lines. So you take a single individual fruit fly that's been mated to a male. We have no idea what the male's genotype is, but there's this one female. She's mated. You let her lay eggs, and then you inbreed those offspring, and then you keep inbreeding over and over and over again. So the siblings are mating. Eventually, you kind of whittle out that extra that male at the original's variance to where you get almost every individual in the vial is near genetically identical.
Interesting. Wow. Yeah, I don't feel like we have anything like that in paleontology.
Well, it's I guess that for like the terminology, right? Because there are some, insects where we have parthenogenesis where they don't need to mate, and I guess we have no idea in dinosaurs if they would have that feature.
We have it in modern birds. It's been observed in condors, I think. So it's not unfathomable that it would be in theropod dinosaurs too. Yeah.
I think would be the closest.
And maybe in crocodilians also. You could use the old EPB test. If it happens in birds and if it happens in crocodiles, then chances are it happened in non-avian dinosaurs too.
I think there were actually studies of it in crocodiles too.
Yeah,
that it happened.
I've got the vaguest recollection of that kind of thing. I think you're right.
Yeah, so it's flies aren't parthenogenic, but it would be, I guess, that similar idea that if you had an avian dinosaur that became parthenogenic, then its offspring could continue to fertilize.
What drink?
Without the need of a male, and then you make essentially a genetic line that's identical to that original one. So it's what you're kind of building towards with these.
Interesting.
So with the flies, it's just personal preference. Scientists like their own. So depending on what paper you read, if you want behavioral stuff, it's this Canton S fly that is from Canton, California. If you want more genetic stuff, it's Oregon R, from Oregon with red eyes. If you want like a Midwest, it's apparently vermilion is the more common one. Why? Who knows? But they are wild type, but they're not identical. So when you're doing those tests, it might be very different.
At conferences, do different fruit fly researchers show up with different T-shirts that have got Oregon red and stuff like that? And no,
it's more subtle than that. It's the reviewers. Reviewers have their favorite wild type,
and they'd be like, "Why didn't you use this one?"
No, they do. They will hammer you, and then there's the other side of things too. It's like if you do a control where you mutate a gene, and so your experimental is you test the phenotype with the mutant, and the control is looking at what the mutation was made in, that genetic background without the mutation. There are folks that will argue that actually the best control is to mate that non-mutant to another non-mutant of a different genetic background, and that gene combination, that's a new gene combination. Tommy, thank you for that gift sub to Reiko. Thank you, Tommy Podicus. If that new gene combination doesn't cause there to be a mutation, then it's a true control. So there's a lot of schools of thought on what a true control is, which is always what I wonder, when you tell us about papers, when you're comparing to for this bone is reminiscent of a two-legged dinosaur, right? And so it's I guess in my mind the control might be we would look back at established two-legged dinosaurs and maybe look at those bone structures and then compare it to the new one and say here are the reasons
you're
why.
right on. That's exactly what we do.
Yeah. And so is there an agreed upon set of controls generally from the research that you do for these kind of things, or is it also kind of is it like the c-elegans model where you have the standard, or is it like flies where it's just split?
It's like a case-by-case thing because you're not going to have one control that works for all groups of dinosaurs or something like that just because we're not going to have consistent information about all of them. There's certain groups of dinosaurs where we just we don't know what their wrists look like. Spinosaurids are like that, where no one's ever found a spinosaurid wrist bone anywhere in the world. And it's just So we're not really sure about what their what the range of motion is in their forelimbs,
Huh.
because we've never found that. So like but like if you talk to tyrannosaur researchers, like different tyrannosaur researchers will have different opinions on like which specimens you should be comparing to which other ones, and like this one I think that's a juvenile, so that's not going to be a good comparison with this guy, and that kind of thing. So yeah. So maybe it is more like your fruit flies example.
Or Smike says that there's just no risk then logically.
There you go. Yeah.
I always sit back and I'm like, I get really frustrated. I'm all on your behalf. I'm like, man, that would be so cool. And like, also, why haven't we found it? Like, how do you? Does it? I mean, because you see this all the time. How is it really frustrating, or is it? You're just kind of at this point.
I mean,
like,
it's just that's just the baseline. It's just how it is. I never think about it. Yeah, it's like how. I don't know. Maybe it's like a person talking to a dog, and the dog is like, you've got such a poor sense of smell. I don't know how you make it in the world. Like how do you manage to get through your day? It's like, well, I don't know. We've got eyes, and we just have different strengths.
I like that analogy.
It's just a different way of looking at things, and it's just very normal. It doesn't feel weird at all.
Okay, I like that. And
Yeah.
Pudge, thank you for the follow. Aaron, welcome in. Smikes was asking, there are cases of mistaking juvenile dinosaurs from completely new species, if that's right.
All the time. Yeah. And it can really mess up a lot of different analyses too, which maybe we'll talk about later when we get to the second.
Yeah,
paper.
that's.
But yeah.
That reanalysis element of that paper I found really interesting because it seemed. I wanted to. We'll talk about it, but like how easy it is to do something like that, and how hard I think it would be to do it in other fields at least.
This is a dinosaur.
Yeah.
This birdie dinosaur. Dinosaur birdie.
John.
Birdie dinosaur. Dinosaur birdie.
Thank you,
Very
JD.
good. Yeah.
So this study, right? Smikes asked earlier too, like, what is the control here in this particular study that we were looking at? And Melia, thank you for the control, right, is this young short generation, and it's a weird control because it's not standard. So they are defining every X number of days we're getting an embryo, and that is laying the next generation, which is then laying the next generation this day, and so on and so forth. So that almost assumes that it's a standardized means of reproducing these animals in terms of time, but it's not. And so here it's almost like there's actually two experiments going on. It's what is the lifespan of this short generation time individual, and then what is the lifespan of long generation time individuals, and comparing the two. Because at least because there is no standardized like when you're supposed to lay the embryos and do the experiments like with a particular age. So like this is frustrating in our field, Danny. We used young flies. Well, sir, what is a young fly?
A couple days, maybe.
No, but right. So, but that definition is.
Oh, I get what you're saying. So you're saying that's an undefined term there.
Like in the method section, it's like
not specific enough. Oh boy.
It's like, well, and if this paper is arguing that there's effects of age on the offspring, then.
And they didn't quantify how old they were?
Well, so in this one they did, but a lot of studies they just say young flies.
Oh, I see what you mean. Yeah.
And it's just so it makes the reason I like this paper is kind of you almost go back and have to consider, well, other studies that I've looked at that say just young or maybe don't even specify, how might the results change as a function of knowing this kind of information.
Absolutely, yeah. That kind of reminds me, speaking of time like that, in the paleontology world, sometimes you'll say, well, these fossils came from such and such geological formation. It's like, well, that might be 100 meters thick. You might have several million years of time within there. Like, where in section in that formation did they come from? Because that'll have huge ramifications for like what the critter looks like, and
you talked about too, like the evolutionary trajectory of the animal. like how a singular animal has evolved over time. If you have millions of years in the strata, there might be beginning, middle, and end differences too.
Exactly.
Yeah. See,
Yeah.
I love that's why I love these conversations because they're so cool. And
Yeah.
Dr. Cat, welcome in. Mama, as a fan, hello. It's Evolution says, what's a great crossover with those cool bird emotes? Welcome the heck in. Sparky, how old is an old human? So that Sparky, these kinds of questions are also very paper specific. And here, they defined old men as over 50 and old women as over 40. Because that's what the studies have looked at, where there's genetic changes with the offspring. So in the very beginning, they had some citations where older, like that's what they define as older, generally considered greater than 40 for women, greater than 50 for men, is that there is data out there for offspring of these aged individuals might have increased health deficits. And that's what they were going for in this study, to try to recapitulate that in flies and the worms. And then if they can make it happen in the fly and the worm, then maybe we can fix it, right? If you could fix it in the fly and the worm, the idea is the genetic similarities exist between human and these organisms, and so maybe you can fix both. So the Smikes, the gametes here are interesting again in nematodes and flies because they have stem cell populations, and so they're producing constantly new eggs. But there does seem to be an aging effect where the ovaries in a fly aren't as good when dividing when they're young versus when they're older. And so you can actually, Smikes, what's cool is there's a genetic trick in flies where you can start aging their ovaries early. So you turn on a particular gene just in the ovaries, and it makes them look like the ovaries of a 40-day-old fly versus a 3-day-old fly. And then so like some chromosome pairing defects that you start seeing in the old fly, you'll start seeing in the young one. Which I think is pretty cool.
It is pretty cool.
Yeah, so the graphs, here are the graphs and like the big crux of the data. And what you see here is the short generation time in the black. Those live about, you're talking roughly almost 30 days until they're all dead. The y-axis is percent survival, the x-axis is age. I have to point out, I don't know what they're doing with their animals if they're only living 30 days. Because, if you take care of them, they'll live for over 60. So it's, I've got a little bit of an eyebrow raise on that. But what you will see is these from aged parents, that's these two blue lines, are significantly earlier in terms of the LT50. So the time at which 50% of them are dead is far earlier. So if we just go along here, 50% death is about 15 days versus you go to the black line, it's about 18 days. So that's a difference there. And then the purple line, that's when you've taken an aged animal and take babies from when they're young that they've been laid. So you do like a short timeline, and then all of a sudden it recovers back the longevity aspect. So it only seems like it only affects them in their olden age, and it can be recovered. So it's a plastic system, which is really, really interesting. I think I have a lot of questions about this kind of study because there's so much stuff about aging in these animals in general, as well as diet heavily influences the ovaries in a fly. Like it actually, if they don't eat, their ovaries shrivel and they start popping, and they can grow back with good food, but there's this element to it. They also buried in the methods, they said when an animal died, they did not replace it, and so there are studies that demonstrate that density affects lifespan.
Oh, interesting.
And there is a difference, Sparky, between male and female lifespans in general in flies. The males die faster, and the females run out of sperm. So this like decreases in fecundity or the ability to lay eggs. Right, they don't tell us like what sex animal dies. It's just looking at total percentage survival, and so it's it's one of those detailed questions, and I'm like, there should be a lot more here, and I'm kind of frustrated that there's not. And then yes, Mike says I'd like to see a follow up with old ova and young sperm and vice versa. Yeah, I think that would be a great combination to have to see, is it dependent on one or the other? Right, for the effects of the parents, is it just on male? Is it just on female? Is it both have to be? Because they only did the both here. There wasn't any other permutation to it. It was just these two old parents have this effect. And then where it gets an added layer of complexity is the epigenetic side of things. So even in nematodes, this has been proven, as well as in flies in a similar context, where if you take offspring from aged parents, those are going to be fundamentally different in terms of gene expression, not just lifespan and fecundity and any of that, but just they are fundamentally going to be different on the gene expression level. So not the DNA sequence, but what genes are turned on and off. And that's true too for nematodes, where they've shown that one starvation effect on the parents lasts 5 generations.
Wow, jeez.
And so you're
That's
dealing
really
with
surprising.
something really complex, where there could be other things factoring in here that's really driving these effects that we don't know of. So it's, in my mind, this is like a neat starting point of a paper, but that's kind of where I'm set on. It's like, this is a cool start. I need a lot more. I don't know. How do you feel about it, Danny?
I don't feel like I'm in a position to make an assessment. It's not my field.
Well, no, but that's I think the cool part, right? Like as a paleontologist, are you convinced by the like? I know it's very different style data, but like, do you find like the argument convincing, or is it just like, you're just kind of like, I don't know, this maybe.
I'm really unsure about it, honestly. Yeah. I don't know, when you're dealing with squishy things in a laboratory, there's so many variables there, and when you're, I don't know, especially after hearing you talk about how, like, they didn't specify, like, how many males versus how many females had died and like, they weren't replaced, and so that also might affect their longevity, because density of the population affects their longevity. They didn't specify what kind of food was provided, that kind of thing. Like, I find all of these a little bewildering. Like,
no, that's a nice sanity check for me too. Is that you also find it bewildering, like that it's not just me. And Cliff's asking, is there an appendix or a link with the raw data that would help answer this detailed question?
Is there sup info anywhere? Yeah.
So Cliff, like things like diet, a lot of these papers just say standard Drosophila media, and that's it.
What does that even mean?
No, that's the thing. That's Danny. That's exactly the thing. No one knows. No one knows what it means, and it's just one of these unspoken things that happens like that. And it's a little bit frustrating that in fact we don't know a lot of these details, and I'm sure it's from you. It's even more frustrating because you're like, hey, I actually don't have a wrist bone for a Spinosaurid. You have the recipe for the fly food. Like,
it's true. Yeah.
And no, Smike,
that's a good point.
This journal is a pretty high tier journal. So aging, in terms of the field of aging, yes, it's a specific like specialty journal, but it's a pretty dang good one. So it's a little surprising that so much got past it. And then with Smikes was also asking about the diabetes in humans during World War. So yeah, I actually mentioned it in the paper too about the epigenetics. See here, even epigenetic mechanisms underlying parental age effect observed in this study are unknown. And in humans, there was this in World War II, the Dutch hunger winter, where this town was starved by the Nazis, and the women who were pregnant gave birth to babies that had type 2 diabetes upon birth, right? And the mothers did not have diabetes. And that's because it turns out during starvation, it's beneficial to have diabetes because of how the sugars are processed. But then...
Interesting. Do you think that's why diabetes originally evolved?
Yeah, it may be. And what's wild, too, is that those mothers, if they had other kids after the starvation ended and they went back to being able to have food, their babies were totally normal again. Their babies did not have diabetes. So the moms were not mutants for it. It was all epigenetic. And then those kids that had diabetes that grew up totally normal lives, their kids also did not have diabetes. So it was
something that was just entirely developmental. It wasn't coded for in their genome or anything. It wasn't a heritable trait in a genetic sense.
But in the epigenetic sense, right? Like, so it's like we've modified something that the genes, like, to predispose you to this.
Finds a way.
Exactly, Tommy. That's how their life found a way, because if they have to be able to respond to these environmental cues and then re-respond, right? So if it was mutation like natural selection, if all of a sudden there's a genetic mutation that
Gucci in the Hoochie Mama.
Exactly, coffee. It's very Gucci to happen that, right? If it was, but you can imagine if it was a mutation, and all of a sudden food resources came back. Now they would be at a fitness detriment. And yeah, JD, 2 bucks would be a great mutation. Danny's never gonna come back. But I just think, it's a neat example of already the natural selection, the ability of animals to respond to their environmental cues.
Interesting.
With those epigenetics. But again, this paper I think is a cool starting point. It shows the same findings in 2 different organisms, demonstrating that they're probably both can be a good model.
But
and
it makes me curious about a few things, kind of just the nuts and bolts of how things work in this field. Is it common for a researcher to work on both Drosophila and c-elegans, or is it like there are separate teams that did these and they collaborated for this paper, or how does that work?
So it's becoming more common to work on multiple organisms because of a funding perspective. Funding agencies want to have more papers and studies and labs that are cross-organismal and demonstrating that there is, in fact, this is a real phenomena.
Right.
It's interesting that this is from France, so it's an EU-based paper. I'm not familiar with the funding agencies in the EU, but it seems maybe it's something similar to that effect as well. But yeah, like when I started in undergrad, that was unheard of, and now many labs are moving towards it. The difficulty is that, and I'm curious about this in paleontology too, because presumably dinosaur paleontology isn't the only field, right? Like there are presumably subsections.
Well, what do you mean? Like it's not the only field within paleontology?
Right.
It's certainly not. The majority of paleontologists don't work on dinosaurs. Vast majority. Yeah.
Right. So like the way you do your collaboration and in your subfield is probably different from a paleontologist who does work in a different field.
To a certain extent. I mean, there's so few paleontologists in general that like saying that there are different factions and it works a certain way over here versus over there. It's like it's almost kind of a case by case thing. There's so few of us in the first place.
Okay.
That yeah. I like to say that there are about as many different ways of being a paleontologist as there are different paleontologists. And maybe that's just because there aren't that many of us in the first place, and we can't. Everybody's already like specialized in their own thing, but they might be the only person working on this particular animal or something like that.
Gotcha. So it's, I guess where it's coming from with us is like in flies, there's a ton of fly researchers, but there are the way in which you collaborate will be different than a worm lab. So there's these inherent cultures in the model organism field of that organism of how you collaborate and how open you are with data sharing. So, for example, like the ants.
Interesting.
In the U.S., there's so few ant researchers. The only way to really get a job is if you have your own species that you're studying. Because it's almost like this lab works on this species and don't you dare touch it.
So there's a lot of like claiming of individual species.
Yeah.
It's very territorial. Yeah, shoot. It's not really like that with dinosaur science for the most part. And there's a lot of collaboration. Like, if somebody mostly works on sauropod dinosaurs, the long-necked, four-legged dinosaurs, they might also dabble in ceratopsians or, ceratosaurs or something like that too. And it's always like a case-by-case kind of thing. Yeah, but then there are some people who like, I only work on this particular taxonomic group. Yeah, and then you have people who are methods people, who it's like they do like biomechanics of, they'll work on sauropods and theropods, they'll work on ancient mammals, and they'll work on marine reptiles and stuff. And then you have certain people who are just they're like phylogeneticists. So they're like only working on really producing those family trees and testing them and that sort of thing. So yeah, there's a bunch of different ways to specialize and collaborate, and it's hard to make generalizations because it's such a small field, I guess.
Gotcha. Yeah, so Ian was asking like, why would you want to work on the same species of ant as someone else? So I guess, Ian, with the fruit flies, near everyone is working on the same species of fruit fly. And that's because everyone's trying to work out the genetic mechanisms, the neuroscience of this organism, and trying to fully understand it and flesh it out. And so if every lab worked on a different fruit fly species, we would not have the discoveries that we would have today that are Nobel Prize winning, because it's dependent on so many other connections that one lab or one lineage of lab, there's no way they would ever have figured those components out. It's like if we all work, there's this one ant species where the queens can become workers and the workers can become queens. And when they become queens, their lifespan multiplies by 6, and they have changes in, the genetics and ovary and all this stuff. Imagine if it wasn't just one lab working on it in the U.S., but you had 6 labs working on it. You might know like how the brain communicates to the ovary and what signals are happening versus just one guy in a lab like slaving away over one question on this animal because it's the only lab that can afford it and has the animal. So that's I think a good way of saying it. Hi, Harissa, welcome in.
Hello there.
And Smikes is asking for you, Danny, is that why? Like, so with dinosaurs, if you're a dinopaleontologist, how much of a selection do you get of picking out what organism you want to work on? Like, or what, like, you mentioned the specialist versus the more generalist researcher. Like, do you have that ability, or do you just kind of fall into what is available?
It depends on what your individual circumstances. So, like, we were watching a video on my channel last week or the week before about one of the only, like, dedicated stegosaur researchers in the entire world, Susanna Maidmont at the Natural History Museum in London, and she might be the only person who like specifically studies stegosaurs, and that's like her thing. And she didn't choose that. It was like she happened to have been given a, I think, postdoc position at the museum, she was saying, and they're like, oh yeah, we have money for a stegosaur person because nobody's looking at stegosaurs, so do you want to be a stegosaur person? She said, yeah, okay. And now she's the stegosaur person for the whole world, basically. Sometimes it happens like that, and sometimes if you're in a laboratory, like you're working under a supervisor, you're allowed to like choose whatever you're interested in, or sometimes they have a specific project, or they want to test a new method, or they've got field work in a certain area, and then you might happen to find a new kind of dinosaur that's really interesting, and then that kind of becomes your thing for a long time. So it really depends. It's like, again, feel like a broken record here, but it's such a small field that like so many of these things are just case by case. Yeah.
Gotcha. That's kind of frustrating.
Yeah.
I mean, it's cool that we have an expert in Stegosauruses now, but that you had to fall into and not necessarily be interested in doing that.
I mean, that is it not the same way in molecular and systems biology?
You have to hunt a position that has funding, and you know, if you're competitive, you can kind of pick what lab you want based on the research question and their funding ability. So it's, you know, if they're funded, as if you're going to get it or not, but you can pick, like, as long as you're willing to move cross country and wherever, it's it feels,
it's interesting. Yeah,
like much more available.
So much of it is circumstantial for paleontology, because it's like you never know what you're going to discover when you go out and do field work, and so you might get, you know, stuck describing a new mosasaur for several years or something like that, and you have to learn everything there is about mosasaurs, that kind of thing. Or maybe you decide not to describe that and you just bring in a mosasaur person and they describe it for you. Yeah, it really depends.
Yeah. I think it gets hairier in the molecular field if you want to do a particular organism, like if it's more unique, like not flies. Like presumably, if you want to look at a fly, there's, you know, hundreds of labs to pick from. But with ants, there's, you know, the 4 in the U.S. that have molecular elements.
That's it?
Yeah, it's because the funding.
4 labs?
Yeah, with actual like funding to do those kinds of experiments.
Wow.
And so you might not be able to do the experiments you're interested in, but at least you can start working on that animal, and then when you get your own lab, or if you get your own lab, you can actually then start working on the question you were originally interested in. But that's where it gets kind of more narrowed down.
Makes sense. Oh, and I was speaking of narrowing things down and like how big these fields are. Just out of curiosity, Bulent, did you know have you heard those authors' names before? Have you read their work before? Are you familiar with the authors on this paper?
I have not. So
there's probably a lot of people who work in this area, like it's in the hundreds, probably. Lots and lots of people.
Aging in general right now seems to be a hot field because we want to live longer, right? And so there's always, and there's so many elements to go off of, where it could be, you know, the ages of the parents, like here, or genetic changes that affect of aging, resulting mutations of aging, the neuroscience of aging, and so there's just an absurd number of it. So there's you get to know people if they're in your very particular specific field, because they're probably going to be your reviewer, and so then, like, you might get a good review, or you might get the bad review. And you probably know who it's from based on their language. For the French, actually, Smikes, we had not collaborators but competitors who were in France for some papers in undergrad who were researching parasitic wasps, and they would send my best friend, the postdoc at the time, nasty messages in French, like via email, to like knock it off. Like, hey, we saw your like poster abstract, like we're gonna beat you. And so it was like, I don't remember the person's name, but Nate would be like, oh, this Jean-Claude fella, he keeps harassing me, or whatever the guy's name was. And it was like, oh man. What do you end up doing? And so you get to know but it's such an open field, and there's, again, I'm not sure in like for paleontology, but there's a lot of incoming people that maybe work on neuroscience or something along that, and then flip back and start working on a question that's maybe very tangential to their work, but now all of a sudden hits on your work as well, just because that's where the research took them.
I mean, we get stuff like that too. And it's like I'll know most of the people in like a given specialist area unless they're a new student, because there definitely is, like, there's this growing trend of like publishing even as an undergrad and stuff like that. which is probably a good thing, I think. Yeah. But yeah, chances are if it's like a multi-author paper, I will know at least one of the authors if it's a paper on dinosaurs. Sometimes I'll know all of them. Sometimes I will have published papers with two of the 5 authors or something like that. Again, it's a small field, you know?
Yeah, it's super reinforcing that. A couple of questions from the chat as well. Reiko asks, is there an issue in either of your fields comparable to the charismatic megafauna issue where some species get disproportionate attention from researchers? It ends up with the possibility of distorting the broader understanding of the given ecology.
For paleontology, there's definitely, well, other paleontologists would say yes, dinosaurs they get a lot of attention, which they do, but we know so little about them anyway that, you know, it's not like we're spinning our wheels over studying certain groups of dinosaurs or anything like that. Everything is understudied still because we just don't have nearly enough people in, you know, in vertebrate paleontology in general. But there are certain dinosaurs you would probably have some dinosaur workers would probably say like, oh yeah, tyrannosaurs way overstudied. They get too much attention, you know, big toothy things. Because they're charismatic, you know, they just get too much attention. And maybe that's true, I don't know. But I can't see how that would like I find it difficult to imagine how that would distort our view of dinosaurs in some way. You know, it's like, really, honestly, everybody's understudied. It's just tyrannosaurs are less understudied than maybe some other dinosaur groups.
Where's my drink?
Yeah.
I like
Black
that.
drink.
I like that analogy. I think it's similar in genetics as well, where there is a charismatic topic, maybe, where it's let's say there's the tau protein in Alzheimer's. So it's this aggregating protein that forms in the neurons of the brain. And for a long time, everyone's like, if you can get rid of that, you cure Alzheimer's. And so all the funding was there, and there was a lot of labs looking at that particular question. And you know, now it turns out you can get rid of those aggregates, but the Alzheimer's is still there.
Oh, okay.
And so it's almost like obsession with this one question, without losing you lose the scope around that particular question. And sometimes the funding also follows it, unfortunately, where you might start asking like, hey, but what about this tangential thing? Or even, I mean, like, there's definitely examples like RNA interference, which we were talking about with c-elegans. So that's how you turn off particular genes of interest in a tissue-specific manner, was discovered in accident in nematodes. And so you wouldn't be able to necessarily get funding for that now because, well, how is that connected to human disease right off the bat? And so I think, like, if there is anything like megafauna like in terms of obsession in human in studies now, it's linking it to humans in the immediate, in the foreground, versus eventually linking it to humans.
That makes sense. People are asking, what are the practical applications for human medicine?
Right away, versus downstream.
Like basic research where you're just figuring out how these things work in the first place.
And
kind of putting the cart before the horse there.
Yeah, and Sparky, so the funding doesn't follow the money. Like, it's not that the funding agencies are trying to make money from this. It's that there's so little funding for basic science now that you need, as Danny said, like an immediate payoff. Like, I need to study a question where I will solve aging with my question right away versus a long-term, you know, maybe down the road we'll figure out something, but let's just figure out how this organism works and maybe we'll find a secret from it.
That's interesting, because I feel like we don't necessarily have that problem as much in paleontology, just because, you know, when we're studying dinosaurs, for instance, that's not gonna cure cancer. Not in an immediate sense, anyway. But, like, you know, it doesn't really have big implications for human medicine or anything like that. So basically everything that we're doing is basic research. We're just doing it for curiosity's sake, and Yeah. But I think maybe that's one of the reasons why paleontology is so underfunded, is a short-sightedness on the part of funding bodies and government and stuff like that. Because, I don't know, I said dinosaur paleontology won't directly cure cancer, but it could indirectly cure cancer, in the sense that there are so many people who end up going into science or into medicine or into other knowledge fields because they were fascinated by dinosaurs when they were a kid. Like, it's a gateway drug into the sciences. Like, dinosaurs are a way to bring people into science. And if you stop funding that, then you might end up cutting off some of the supply of, like, people going into the scientific fields. So who knows, if there ever is a cure for cancer, you know, whatever that means, cure for cancer, it might arise because, you know, some person got into science in the first place because they were interested in dinosaurs, you know?
Yeah.
There's actually, did we talk about this last time? I can't remember, Bolin. But I really want to, like, actually do a study, like, take a representative sample, big survey of, like, people in different knowledge fields, not just science, but also, you know, medicine, engineering, maybe, like, I don't know, law or something like that. Just ask, like, how many of you were obsessed with dinosaurs as a kid, and that's, like, how you learned how to, you know, fall in love with learning. Because I really think it is, like, probably a significant number.
I mean, I know
Certainly
I was.
non-zero.
I was obsessed with them.
So
And
I'd really love to
finding
be able
your
to actually
channel
commission a study like that.
Well,
So, yeah,
it was finding your channel that rekindled that love of dinosaurs. Because it's I don't know who all in chat has experienced this too, but you're told, well, you can't make a career out of that. Like, knock it off. Almost, you know, and so that's I know, like, I've undergone that. I know some of the friends in the community have also undergone something like that, and so it would be accessibility to, you know, seeing people succeed in it. Even if you don't end up becoming a paleontologist, it's you can be curious and follow a life of curiosity. I think would be like a good. I would assume that many people who you would ask would have been interested in them, and by asking that, they'd be like, you know what, I still am.
Yeah, there you go. Yeah. So, I don't know. I'd really love to actually be able to get some numbers on that at some point. And, yeah, I don't know. That's the best way that I can think of, like, how to improve the funding situation for paleontology, is to maybe just get some people to realize, like, hey, shoot, this has much broader implications for science and learning in general. You know, you fund dinosaur science, and you're indirectly funding, or indirectly it may be helping to make scientific advances decades into the future, if you're inspiring young people to go into science in the first place.
Yeah, I think based on some of the questions in the chat about having studying things for the sake of studying them, just out of curiosity, maybe our next collab we can. Would you be up for looking for finding, like, an old paper, like, you know, that clearly goes, like, I've got a couple in my head already where it's like, there's no immediate human health or anything, but it's clear they're just having fun for the sake of discovering something. I think it'd be kind of cool to look back at some of those papers for maybe our next collab.
It'd be tough to find one in paleontology because that's almost everything that we do. But yeah, like, from some other field, you know, like, I don't know, playing classical music to oysters or something like that, or I don't know. I'm sure we can find some interesting stuff.
One of my favorites is taking fruit fly larvae and doing tissue transplants. Like, snipping out a gut cell component and transplanting it into a different larva. Number one, the methods are so poor that you can never replicate it. Because like we cut it open and we did a tissue transplant. Sir,
Nice.
No one today can do this because it's like the fine motors, right? It was just, but it's from the 50s. Like,
oh, was this the one guy you were talking about who, like, he was the one person in the world who actually figured out how to do this by hand?
Yeah. He and his wife like did all these experiments, and it was like you just read the papers, like they're really just having fun. And
Yeah.
it was, and it, but it like they were clear, like if you transferred this tissue from one individual to another that were distant relatives, there would be an immune response against it, and they figured out self-recognition and non-self-recognition in the tissue organism. And Travel, thank you for contributing to our microscope camera fund. Thank you for that, Travel.
Very
We're
nice.
looking
Excellent.
to upgrade one of the camera systems that we have for taking pictures of the amber.
Nice,
Thank
nice.
you, Travel. But yeah, do we want to move on to the next paper? I know we could get wrapped
Sure.
up in all the questions as well. I'm always having a blast chatting with you.
Yeah, yeah. I'm having a great time too. Yeah. Yeah, should we move on to the Troodon one maybe?
Yeah.
Or the dinosaur isotope one? Yeah.
So I've got the paper and I've got the thread as well. Whatever you'd like me to pull up first.
Cool, yeah.
The Tradoon. The Tradoon.
We
The
can just do the
Tradoon.
paper.
Okay.
Yeah.
Yeah. So I.
What did you think about this one? Like, what were your initial thoughts?
The Tradoon. The Tradoon. The Tradoon.
I had difficulty with the methods. To be
With the
totally
methods,
honest.
interesting.
Yeah. So like, what they were doing in terms of like getting the isotope readings. Flying Spaghetti Monster, thank you for the follow. Welcome.
Oh, we're honored to have you here.
It felt like the composition of these minerals and like what all is in here should be. It's almost like the mass spectrometry equivalent experiment in molecular biology, where you get a measurement of like all the proteins composition and amounts that are inside of this sample, and that's what you use to interpret like whatever hypotheses you're having. And so it's just, I guess, how the elements that they're extracting from the teeth relate back to the diet, which then relate back to if they were herbivorous or not. Like, I was having like connection difficulties with that.
Yeah, which is like how the method works and why it's reliable. So I'm maybe not the right person to ask about the specifics of the methodology there, because like I don't work on isotope stuff. There are other paleontologists who specialize in this, and like that's their thing. But this is a methodology that archaeology has actually been using for a long time. So like looking at the diets of, ancient peoples by looking at their teeth. And like it's a pretty well established method there. And it's cool that we can now start to apply this to dinosaurs too. But and that's fairly new there also, that they've been able to actually find stable isotopes that you can use this on for dinosaurs. But from my understanding, this is like fairly typical stuff. Like it's almost standard to the point of like being boring in archaeology.
Okay.
This is like a very standard method. Yeah.
Is it? Do you unless I misunderstood, which is very possible, right? Outside my field, they don't look and compare to modern day animals and their diets and look at the isotope readings and like try to like have a comparison of what is herbivorous versus what isn't, because this almost feels like a big change in what the Troodon used to eat. And so
I do think that they were actually comparing that to, I don't know if they actually stated explicitly in the paper, but I'm pretty sure that those particular isotopic signatures, you can see them in modern animals, and like that's settled.
Gotcha.
Like there is a standard for that, I think. Yeah.
Okay.
Yeah. And the cool thing is, maybe I should have put this in the email beforehand, but for decades there's been this idea floating around, really speculative, that like, oh yeah, maybe troodontids did eat some plants too, because the shape of their teeth, where they've got these big old serrations on them. They almost look like serrations on a leaf or something. And sometimes you'll hear troodontid teeth being referred to as leaf-shaped teeth, which is consistent with plant-eating animals. Critters like iguanas and stuff like that will sometimes have similar teeth if they're eating things like leaves.
This shape,
so there you
right?
go, yeah. So those big old serrations, like these are huge denticles there. Denticles are what the things are actually called. And this looks really similar to a lot of plant-eating dinosaurs, which is weird because this is clearly like a theropod, and it it looks very raptorial overall in its body shape and its claws and everything else. And like all of its relatives are all of its close relatives, the other Deinonychosaurs, are all predators. So this idea that like these guys may have been munching on a lot of plants too, it's an idea that we've had for a long time, but no one's ever actually been able to test that. And so it's really cool that like in this big sample of different dinosaur teeth from this geological formation, that these guys, they kind of show up between the meat eaters and the plant eaters. It's kind of like, it's a nice vindication of that idea, I guess.
Where did the idea originate? Just from looking at the teeth?
Just from the morphology, yeah. Just
Okay.
from the shape of the teeth.
And the proving now is by looking at the isotope signatures.
Exactly.
So when there's a proving like this, do you have to get multiple populations? And again, this is my naivete of knowing how many troodontids we have and like where they're coming from. But
this has only ever been done once, I think. I think
Okay.
this is the first time. So it's like, this is a cool start. And it'd be cool if we see this same kind of pattern in other populations as well, other troodontids, other environments, other geological formations. But yeah, it's
so we
this is
don't know
the
if it's
first study of its kind for this.
if these are just special compared to the other.
It could be. It could be that these guys are just. Yeah. We're not totally sure yet, but it's tantalizing, I guess.
Yeah.
You know,
No,
it's exciting stuff.
It's super cool, and it's. I mean, but that's the neat thing about that kind of answer is that either one, it doesn't matter what the answer is, right?
Yeah.
Because if it's a unique troodontid that was,
That's still cool.
herbivorous, why, right? And if it's not unique,
Yeah.
well, that's
Yeah.
cool too. And that's my favorite kind of science, right? It doesn't matter the answer.
Yep. And there are, you do see this in like different populations of animals living in different places. They might have the same morphology and like totally different diets. So like I think bears are a good example of this, where like you might have grizzly bears living in one environment where they're eating mostly roots and tubers, and bears in another environment are eating a lot of salmon and stuff like that. And they might be fairly identical in terms of their morphology, like their teeth might look exactly the same and their skeleton and everything else, but like it's a different lifestyle. So it's kind of, it's almost a cliche with theropod dinosaurs, with the two-legged, mostly meat-eating dinosaurs. It's almost a cliche nowadays that like, oh well, this overall body plan, being a dinosaur walking on two legs, having an S-shaped neck and all that, and four limbs that you can use for different things. This is a really, really versatile body plan. You could do all kinds of different stuff with this basic body plan, from being like a big two-legged hulking predator like Tyrannosaurus to being a creature that's essentially like an insect, like a hummingbird, that's still got the basic theropod body plan. So it's like a really, really versatile way to have your body arranged. And yeah, it was just kind of further highlighting this. Is like, oh shoot, troodontids, they may have been partly herbivorous. Theropods just get up to all kinds of different wacky stuff. It's such an all-purpose kind of body plan. Yeah.
I think there's two questions from the chat. So from Hiptacularaptor, if I'm hip, I'm adding a little. Please correct me, but if we're taking these different troodontids that are maybe having these different diet plans and maybe specializing on these different food, maybe I'm getting out ahead of ourselves, right? But is that maybe how you would start defining subspecies eventually?
Subspecies are such a specific thing that like we probably won't be able to recognize those in the fossil record, because they're just too similar. Like when you've got different subspecies of insect or something, we don't even really have a hard line on what a subspecies is. We basically don't have a hard line on like what a species is in a lot of
I think a lot
contexts.
of fields now are having a lot of trouble with just the general what is it, right? Because like originally it's reproductive barriers, and now we have grizzly bears and polar bears mating because of changes in climate. And are they still a different species?
So yeah, but maybe if Hiptacularaptor meant is this how you have lineages split? maybe that's what they meant. Then, yeah. In fact, this sort of thing might be going in the opposite direction, where like having a like really versatile teeth like this, where you can eat plants or meat, you're just kind of a generalist. That might be kind of a safeguard against speciation, against a lineage splitting apart. Because it's like if you can eat plants or you can eat meat and you could just move around and do whatever you want, then it's not like oh, this population over here only eats plants and this one only eats meat, and that's why they eventually become two separate populations and two separate species eventually. It's like the more of a generalist you are, the less likely it is that you'll have populations that will like speciate. Maybe, you know. We're kind of getting off into the weeds here and getting really speculative, but yeah.
I mean, it's kind of fun though, right? Like, so Hugan asked, would that be there be a possibility that most dinosaurs, if they had the chance, maybe leaning on omnivory versus being a specialist?
We're starting to uncover evidence of that. Yeah, that like, well, last week when we were talking about coprolites, about fossil dung, it turns out that like duck-billed dinosaurs, in their dung, there seems to be a lot of like rotting wood, which is really strange because it's not like a nutritious thing to eat. And so Karen Chin is a coprolithologist. She studies fossil dung, and she's figured out, she thinks that they're actually like rooting through rotten logs and stuff like that to get to invertebrates inside, to get the extra protein before they lay their eggs. Because all of this dung with the rotting wood inside was found near dinosaur nesting sites. Mm-hmm. So these big duck-billed dinosaurs, it's like they've got to ingest all this protein however they can before they lay their eggs. And so, yeah, they're like almost doing something kind of omnivorous there. And maybe they would chow down on a little mammal or something if it ran in front of them. And who knows, you know? But it's interesting because it's not something that we see in modern mammals, really. It's like, do you have great big herbivores like elephants walking around looking for insects to eat? Like, they just don't do that. So it's one of those things where a lot of our assumptions about dinosaurs, like, oh yeah, well, they must have acted like elephants, and elephants don't do this, so they probably didn't do that. So, no, those are sometimes bad analogies. Dinosaurs were just weird and different, we're finding out again and again. Bizarre creatures unlike anything that we really have today.
That is a really cool finding about the dung.
Isn't that neat?
Because the possibilities it opens up as well, because you're getting behavioral information potentially, the diet as well. You know, my brain went to maybe if there's some kind of endosymbiotic relationship they have that maybe helps at some level break down the wood as well, like in their gut microbiomes. Obviously we can't test that, but that would be like an additional, like, why do they do it versus other dinos maybe didn't. Maybe it's not just for the invertebrates, maybe there was a little something else there, and that's a really cool finding.
Yeah. Isn't that neat? Yeah. So I wonder if you could like test any of this stuff isotopically in the future, look at the teeth of these duck-billed dinosaurs and see if well, that was an interesting thing too in the same paper, is that like they actually show a higher than you would expect level of protein intake for being herbivores, which is kind of unusual, you know. It's not like a bison or a cow or a giraffe or an elephant where they're pretty much only eating plants.
Yeah.
So yeah, it's interesting stuff.
Yeah, it's and that presumably is from the meat eating element to it as well that they're doing, right? Mm-hmm. Not just gaining from a particular plant.
Probably, yeah. I don't know. I don't really work on isotopes. Not really. I don't at all. So, yeah. I'd love to be able to talk to the authors of this paper. By the way, I think it was Thomas Cullen is one of the authors on this, and I used to run into him all the time out in the field. Because his graduate advisor at the time, Dave Evans, ran a field crew back in Montana in like 2015, 2016, 2017. We used to run into them all the time in town. And sometimes we get dinner together and stuff, talk about how the field work is going. So again, it's a small world. Like,
Yeah,
I know some of these people personally.
that's really
Yeah.
cool. It's funny. Also,
Yeah.
several people in chat had the same comment of like, what we define as herbivores will eat meat when given the chance.
That's
with,
true.
With
Yeah.
different examples. We also had a question from Quiglius wondering if we look back, we look back and criticize on early scientists' observations, theories, and discoveries, maybe how they got it wrong, which actually will dovetail nicely into another paper tonight.
Yeah.
Do you think that people in 100 to 200 years from now will do the same thing of us today?
Oh, 1,000,000%, yes. um, Definitely. Yeah. Yeah. I mean, shoot, we do this today for scientists now, like with brand new papers. So, of course, people will do that in the future. Yeah,
Yeah.
absolutely.
And I think with
Yeah.
the technology changing and evolving over time, it's almost making it ripe for such an event where we can have more and more criticism. But I think valued
Oh, yeah.
criticism where it's gonna be like, for example, like we can do single cell RNA sequencing now, so you can figure out in a single cell what is the RNA expression, like what RNAs are up and down compared to the neighboring cell. But it's only so good in doing the highest population of RNAs. It's like maybe the top 300. Anywhere lower in the weeds, the tech isn't good enough to discern what's background noise and what's actually different. And so,
But in the future, that technological boundary will be transcended.
Yeah.
Like we'll have better tech in the future that could do that.
I meant like 10 years probably will be better tech.
Yeah.
To figure that out. And like with dinosaur excavation, I know you tell us this on your stream too, and it's always so disappointing that like it's not like sonar, right? That you can't just like figure out where the dinos are and know where to dig.
Yeah.
But do you see there being some kind of tech like that in the future where
not for finding fossils necessarily, but for analyzing them, certainly. Like isotopic analysis is something that's like kind of almost brand new for dinosaurs. Like it's not been done for more than like maybe there was one laboratory that was starting on that like 15 years ago or something, but it's pretty new stuff, and we're going to be seeing this like more and more, and hopefully in the future it'll become kind of standard. That'd be really cool to see. But yeah, in terms of the actual excavation process, I don't see technology helping with either finding or digging up dinosaurs. Yeah. You remember the ground penetrating sonar thing from Jurassic Park and that line, like, a few more years development, we won't even have to dig anymore.
Yeah.
It's like, that was 30 years ago. That was 1993. And it didn't work then, and it doesn't work now. So
is that a function of the tech or the soil or both?
It's not the tech. It's just that the fossil bones themselves and the rock are essentially like the same density. And so it's like a fundamental mechanical problem there, where sound waves are going to travel at the same speed between both of those. Yeah, there's no way around it. There are some places where you can actually detect dinosaur fossils, or fossils in general, I guess, with radiation, because they will be slightly more radioactive than the surrounding rock, because when they were being permineralized and fossilized, there was like uranium in the groundwater, and it like collects in the bones. But I don't know if that will ever be an actual workable way to find dinosaur fossils in the first place. Because the thing is, if there's something under the surface, you still have to dig it up to see if it's any good. And it's much more efficient to just kind of walk around and look for interesting bits of bone sticking out, as opposed to just, not every dinosaur fossil that we find is going to be something worth digging up. Sometimes you'll have garbage ribs and stuff like that. It's not worth bending down and looking at for even a minute.
That is also fascinating to me, of how you can tell, like, what.
Yeah, it takes a lot of practice.
Yeah.
Yeah.
Because naively, I think every single thing you see, but I know like your time is limited, the funding is limited.
Yep.
But it's like, what if connected to that rib there's that elusive Spinosaurus wrist bone? Right.
I mean, yeah, anywhere that you step in a given area, there might be the world's greatest dinosaur skull just underneath your boots, and like you'll drive yourself crazy if you just keep thinking about that, you know?
uh,
Yeah. You got to go with the sure thing, so like walking along and actually looking for stuff poking out.
Gotcha.
Yeah.
Literate Ian was wondering about the tech that we use to 3D model the pyramids. So I guess like when you do the scanning where you can see like, the internal chambers and like how those chambers interact. So even
That works because in archaeology you're dealing with soil instead of rock. It's like a fundamental difference there. So and with the pyramids there's different densities to, you know, these big limestone blocks that they were using as opposed to the surrounding soil and everything. Whereas with, you know, dinosaur fossils, they essentially are rocks. Like,
yeah.
There's sometimes original organic material in there, but like the bones themselves are going to be generally the same density as the surrounding rock, and if they weren't, like, how can you tell from a fuzzy image on a screen if it's like a piece of fossil wood that you're looking at, or if it's a dinosaur femur? You know, like
It would
without
just be
actually
rock on rock. Yeah.
exactly, yeah. Even if it wasn't rock on rock, still, yeah. The whole thing from Jurassic Park at that dig site scene, that was pure fantasy, unfortunately. Yeah.
Does that apply? Do we know if that applies to oceans as well? So that's what Pinky's wondering.
I'm not sure if I understand. Oceans.
Let us know, Pinky.
Like,
maybe like scanning.
Yeah.
I know part of the ocean scanning issue is just the depths, that the amount of time for that sonar to come back is that you start losing resolution, so it's not necessarily even the permeating the soil depths to seeing what's down there, but it's just like your working distance is a limiting
that and temperature
factor.
gradients as well also can play havoc with that, from what I understand.
Yeah.
From World War II submarine movies.
Yeah. Nelf made a cool point, is if we had a program app similar to iNaturalist to look at either aerial photos to ID spots to look at for fossils, or even maybe going on those lines of like, you know, with iNaturalist you can take a picture of a bug and it'll tell you with the predictive AI what it might be.
Well, and that's because the bugs are like, everything's kind of clustered around a mean. For a dinosaur fossil, there's just too many variables. It's like you've got an animal that might be the size of a school bus. It's got, you know, 280 bones, and they can each, you know, be photographed from a million different angles and be broken in a zillion different ways and everything else. It's just like you would never get a large enough training data set in order for it to be useful. Like, that sort of technology would be used in hundreds of other fields before it would ever trickle down to dinosaur paleontology, I guess.
And it's probably
that makes
like
sense.
you said, the number that you have available, like the replicate number total, presumably is not high enough. And then, like, I guess, thinking of the breakpoints, I didn't even think about like how even if you had hundreds of samples of how they're all broken.
Exactly. And if you're looking at for dinosaurs that nobody's ever found before, like, all that's going to be new anyway. So yeah.
And like, the OG Mountain has a cool question too of like AI assisting paleontology. Like, I know in our field, if we make the comparison of the, you know, these like the isotopic signatures, that could be something that could be a predictive metric of what the animal may have eaten as well. Like, you can use AI to predict those elements to it, so it can be built upon, I think, the existing technology. But I guess
so.
What do you think about that, Danny? Is that like, what do you feel about that?
It seems a little far-fetched, just in that so much of what we're doing in paleontology is brand new, and like completely. It's like we're dealing with animals that nobody's ever seen before because we're digging them up for the first time. And from what I understand, AI is good at situations where it's got a lot of training data, and if you don't have any training data for it, like, it can't do anything. And so much of what we're doing is brand new, where there's no precedent for it, that yeah, I don't know. I could maybe see AI. Maybe AI is already being used in making phylogenies and character states and stuff, but that also is such a fiddly thing, and it's such a case by case kind of deal that I struggle to think of any way in which AI could actually help in paleontological research. Yeah, I don't know. I'm also just kind of skeptical of AI in general. I know that's like almost kind of can't say that sort of thing on Twitch because people are so tech savvy here, but
That's how we're getting recommended viewers, Danny. We need the AI.
exactly, yeah, I know, right? Yeah. But I think a lot of AI stuff is way overhyped, honestly.
Yeah, like in biology and the molecular side of things, it's predictive to maybe how a protein functions, but like you said, that needs a lot of base material that it's working off of.
Yeah.
Like
You've already got a lot of numbers in there that you can feed into the model.
Yeah,
whereas
like,
we don't have that for dinosaur paleontology. Yeah.
Gotcha. For us, like even one of our papers is based off the idea of machine learning. Like we gave this algorithm these known learning and memory genes that have been proven in the past, and it looked at sequence changes of those memory genes and then saw what else behaves in a similar way, and it was like, hey, this histone deacetylase behaves in a similar way, you should test it. And we tested and it was like, sure enough, it does. But it would be it would be basing off, like, you're having to have that workable data set, which is why the only example I can come up with offhand was like this paper where if you have these isotopic readings from modern day animals, like a whole, a mass of them, and maybe you can compare and have some kind of number crunching to go back.
Yeah. With this, it was pretty simple. It's just like, well, how much protein is in the diet? Where I don't know if dinosaurs would be too different from modern animals, where you could be like, oh, well, this is a perfect match to, like a giant African ground pangolin. It's like, well, you're never going to have that kind of a match in the first place. So I don't know. Yeah.
Is that because the dinosaurs would be so different, or because of the resolution of data we don't have for the pangolin?
Both, actually, come to think of it. Yeah, because a lot of this basic research hasn't even been done on these modern animals.
So that's what I guess confused me in this paper. Of like of how they got the connection back, because I wondered. I mean, like, what?
I guess it's. I think maybe something you're getting hung up on is that you're thinking it's more precise than it is, where it's just they're kind of looking at like, well, this one's got more of a protein signature in its teeth than that one, and it's like the most basic of the basic here, where it's like, okay, you could do the same thing looking at the teeth of medieval European peasants and see how much protein they had in their diets by looking at stable isotopes in the teeth. It's not saying what they were eating. It's just like, oh, well, these have a higher protein signature. That's basically all it is. It's pretty simple stuff.
So higher
but
protein,
it's like groundbreaking for dinosaurs. Like it's really exciting that we can do this kind of thing.
So higher protein signature plus the shape of the teeth.
Yeah.
Gotcha. Yeah. Okay.
Yeah. Well, I guess for Troodon it would be a lower protein signature. Like for whatever reason they're not getting as much, which maybe it means they're eating plants. I think that's the general gist of it.
Okay. Gotcha.
Yeah. I'm oversimplifying a little bit, but not that much, I think.
No. This is why I was like, I think it's, again, the differences in the fields, right? Of like, well, what am I missing here? Like, no, you're not missing anything. This is
you're overthinking it. It's very, it's almost, simple to a silly extent, because like we don't have that much information in the first place. We got to use what we got. Yeah,
I guess it's like, it's just well, maybe it's not surprising that the modern data is also so poor, right? Like that we don't have the
you'll find that again and again with modern animals. It's like you might have their genome and everything sequenced before you even have a paper describing their skeleton and saying how many bones they have in their neck or something like that.
Yeah, which seems kind of weird, right?
Yeah. But I think it's because that's like I don't know if I'd say it's simple, but that's like a high yield kind of study that you can do, is like sequencing a genome and like figuring that out. But you might not even have data on like what those animals are eating in the wild, or like what time of year they breed or something like that. Like, it's astonishing what we don't know about animals that are just walking around outside right now at this very moment. It's
I
like,
have all right. We should be.
all right, are we back?
All right, we're back. Excellent.
All right.
Thanks, y'all. Sorry
Nice.
about that. There's it's not the joys of a live stream, right?
Wouldn't be live if there weren't some hiccups to it.
Well, I don't like you were talking about. I think today, Danny, talking about how important it is to be here on Twitch versus YouTube. That we can have live interaction and chatting with people, and I think it makes the world of a difference to be able to do something like that versus just pre-recorded and not have the dialogue.
It really does make it so much more engaging, I think. Like, yeah, and I think there's like an added level of transparency when you're a scientist and you're communicating with people live and they can ask you whatever questions they want. That feeling of openness, I think, is really, really important, especially nowadays with so many people, being kind of like actively hostile to science. Nobody in this community would do that, obviously, but I mean, yeah, it's important to show that kind of openness, I think.
And part of that stems from just the inaccessibility of some scientists, right? Like the not wanting to chat with people because scientists are above them in some way or something. Like, I know that's my last.
Why would you condescend to the hoi polloi? Of course.
It sounds like my last PI. I think the anger is justified from people when that's the attitude.
In some cases, yeah.
And so
when I did a talk for the Society of Vertebrate Paleontology recently, talking about like how scientists used to be some of the most well-known members of the public. Like, some of the most famous people on Earth in the early 20th century were scientists. And then the Cold War came along, and suddenly scientists got all of this research funding kind of poured onto them by the defense industry. And they were told, like, don't interact with the public, stay in your laboratories, and we'll continue funding you. And so at the same time that, like, the public is becoming increasingly wary of science because of things like nuclear weapons and stuff like that, also, they don't have scientists interacting with them anymore. And so it was kind of a perfect storm of it was just a bad situation for scientists to be in. And then at the end of the Cold War, the funding dried up too. So scientists didn't know how to talk to the public anymore. Or, like, there was a culture in science of you don't talk to the public. And there was no more funding. So it's the worst of both worlds there. And
and that culture is sometimes still pervasive in a lot of fields.
It really is. Yeah. Thankfully, I think not so much in paleontology. Largely because we never really got that much defense spending money in the first place. We always kind of, to a certain extent, had to go out hat in hand to the public and say, hey, what we're doing is cool. Can we have some money for this and that? But I know in other fields, there's definitely, like, physics, I think, really suffers from this. or like so many people are deeply distrustful of physics in the general public. And they're like, Large Hadron Collider, that's going to destroy the Earth. Let's, what are these scientists up to? That kind of thing,
I mean, we get it in flies all the time. Why are you studying bugs?
Oh, yeah. Uh-huh.
And it's the inability or the lack of desire to want to interact with the public.
Uh-huh. Yeah.
Gojira has a boring methodology question. Go for it, Gojira. It's not going to be boring.
Okay. Yeah. Well, and I think maybe also people in genetics labs are usually not going out and doing much field work, right? So, like, most of the time is probably spent in the laboratory. Kind of like, indoor people. I think, not to, like, bring stereotypes into it, but, like, I'm not saying, like, oh, paleontologists, we're the jocks of the science world or something. But, like, I do think having to go out and do field work, I think, does maybe give you a sort of a different mindset about the way that your science works also. Like, if I were stuck in a laboratory all day, every day for my whole career, I might have kind of a different perspective on doing things like outreach and stuff like that, Because the way that I do field work right now, it's like we've got to have volunteers on our crew. We're constantly interacting with new people. You've got to interact with the landowners and government officials, and like you've got to maintain good relationships with folks, and like sit down at the end of the day and share a beer with them, or like cook up some burgers and that kind of thing. There's like I think there's a general need to like be personable in a way that like maybe isn't always there in the laboratory. I don't know. Does that make any sense? Or
Oh yeah. It's like,
am I off base here?
It's the difference of having an echo chamber versus having to engage with others outside of your field, even like for not even
like regular folks.
Or even collaborations, right? Like you're not just everything you need isn't in one place.
For sure. Yeah, that's actually a really good way to think about it. Yeah. I mean, like if I want to study a certain group of dinosaurs, I might have to like fly across the country and go to a museum and like make friends with the collections manager there, and like it's not as insular, I think, as some other scientific fields can be.
Yeah.
It requires more collaboration, more interaction with other human beings.
Yeah, I was lucky to do some field collections in undergrad and in postdoc, but it's not. That's, I don't think, definitely not standard in the molecular sciences. It was more, and it wasn't like collecting fruit flies necessarily of the same species. It was like, hey, we're studying these other species, so let's get more of them.
Yeah,
that was more.
Are there stereotypes maybe in your field about like field scientists? Like, oh yeah, those guys are crazy and they're just out there to go out camping and be wild and rowdy, or are there stereotypes like that?
I don't know of any for like field researchers, but there's more some stereotypes on the model organism that you study. For example, the fungal community is known as like, oh bro, how's it going, man? Like that kind of vibe.
Okay.
And so like there's some of that is like the but no, like for field researchers, that was more the only thing I ever heard about field researchers was like, oh man, like how hard they have it. Like it was more of like a
really
like good thing we're not
Fieldwork's
in the
a
field.
joy usually. I don't know. At least in my field it is. Like I live for that. I look forward to it all year round.
I think maybe it's the funding situation difficulty of like we don't. What could we like like if you're working with RNAs or something, like you'd be running back to the lab to preserve the specimen. It's like the difficulties of working with it, and I think that's one of the hard parts of like the fear of almost being a field worker. Or like when we collected ant queens in the field, it was like, well, shipping them back was a huge like I don't like you've told us how hard it is to move fossils, but like the number of permits that we needed to ship an insect from Florida to Pennsylvania was great. And like because they're living, if it gets too hot or cold, then they're all dead. It's like
and if they get delayed in shipment, like they might just die of old age on the way there. Probably not
Yeah.
with ants, but yeah.
Well, we
Fruit
had
flies, maybe.
there's also like when you can ask different labs for fly lines. So if someone's made a mutant that you need to use for an experiment, you can usually order them from a stock center, but if they don't have it, you can get it from the lab that made it, if it's the lab is still around. And so we contacted this lab in China, and we're like, we really need these flies to do this particular experiment. And they didn't have like the right export licenses or what have you. And so the way we ended up getting them was they sent us a Hello Kitty that they cut into a big plushie, put the vials into it, sewed it back up. And then when we got it, we to cut open this plushie to get the.
That's really funny. It's like a smuggling drugs or something like that.
Yeah.
That's really funny. Wow.
Gojira was wondering, do you think research focused on a specified applicable outcome risks narrowing the possible gains of that research by predefining the target, limiting the researcher's ability to capture unexpected data, broaden the field's knowledge?
I've got opinions on this.
I do.
Yeah, but I'm eager to hear yours because it's a question that's more relevant to your field than I think to mine.
In the worst of ways, we, because of how the funding works, it's almost predicated on when you submit. I don't know how it is in paleontology, but when you submit a grant in molecular biology, half of the grant is actually already done, but you submit it as if you have barely done any of it.
Yeah.
Because that is the kind of time frame that you're working with. And if you're then thrown for a loop, it can destroy the possibility of you getting further funding. So it almost incentivizes you to look through a lens to force an answer that maybe isn't there.
Yeah.
And it's really upsetting to me because it almost creates a situation, or almost like a culture where the outcomes are decided before the experimentation starts, or you set up the experiment to get a particular outcome versus just like, I wonder if this phenomena exists, let me just test it. Right. That's not how you're going into it. You're going into it with already looking for an answer. And that Gojira, unfortunately, that does happen. Like even this paper that we were looking at tonight, right? Like with the aging, I gave y'all a lot of examples on how I think the results could change if you just did the experiment slightly differently. Now, I'm not saying that's what they did here, but it may very well have been that if they did 5 pilot experiments, right, like tests, and then they do, they see which one kind of gave what their existing hypothesis was, and that's what they go with. Then we have a penguin who's upset here. But is that actually something that's then representative of the field and like what you're studying, or is that a contrived experiment that isn't really meaningful?
Right. Yeah.
Yeah, Danny, you said you have thoughts on it, so I'm excited to hear what.
I think. Well, I don't know. I think you and I have spoken before about the grant application process and all of that, and how that often does a real disservice to science. I think that's part of it, and like you're expecting to get a certain result, and so you're more likely to get it, maybe in ways that aren't ideal. It's interesting to I've never thought about this before, but we don't really have that much in dinosaur paleontology, maybe, for instance. And that, if we're getting funding to go out and do field work, it's like we're either going to find stuff or we're not, and there's not really a way to fudge that. There's not a way to find something even if you didn't actually find it, unless you're just misidentifying things, which I guess sometimes happens. But our it's not like our funding is predicated on us finding a certain kind of dinosaur or something like that. It's like you go out, you get funding to go out and do field work, and you find what you find, and it's a little bit more descriptive rather than prescriptive in that sense.
So
And as for
yeah,
go ahead.
There's not like I guess the only thing I can think of is like no one's gonna put dinosaur bones at an existing site, right? And be like, oh,
Yeah.
I actually found a great site, even though these are from, you know.
Yeah, yeah. Yeah, there have been some scandals like that in paleoanthropology in the past, way back in the olden days, like with, I think it was Nebraska Man and also Piltdown Man and stuff like that. But that was a long time ago. It's like Gilded Age stuff from like the late 1800s. Or maybe it was a little, I forget the exact dates. But yeah, I can't really think of any examples of that in dinosaur paleontology. Yeah. And there might be field work to run phylogenetic analyses and stuff like that, like funding to do work in the laboratory. But generally, it's like our science is really guided by the fossils, and they are, you could almost say, set in stone. So there's not a lot of tweaking them or fudging them, I guess. Yeah.
I guess one question I have for you on that, and Gur's probably like, Gojira's like, why you're like going so rabbit hole off my question? But if there is like in the behavioral sciences, like with the insects or what have you, if you come up with a contrived experiment and you get this certain behavior that you only get in this one situation, but you can use it to study important questions, does that, I don't think that necessarily devalues the science of it. It may be just, it would make it so maybe you're not getting ecological information about it, but
Sure.
maybe you could be using this contrived behavior as a tool to study something. And I think that's sometimes an important delineator that's not always addressed, is like, like this in the wild is never going to happen, like this paper.
Right.
And so like we can't get any real information on that front, but maybe it could be this weird tool that we've built to study a particular question.
Maybe it's kind of like with the you are familiar with the paddlefish sturgeon hybrids, right? That they made. So like that's not something that's ever going to happen in the wild, but that's like a really interesting thing that you can do in a laboratory, and you can use that to study certain phenomena. Is that kind of what you're talking about?
Yeah, like maybe like that's one where it's so like Gojira, correct me if I'm, but if you're like having this predisposed idea of like the result that you're like looking for, and so maybe the result is I'm looking for a particular behavior, and I've made it this contrived series of experiments that'll never actually happen in the wild, but I'm not describing the behavior as this big discovery, but rather the outcomes from it. Like I made this behavior paradigm, but now I can test like memory components or the neurosystems behind it, and it's just a tool.
Well, isn't that how most experiments are? Like with the rat imagination one from our last crossover.
Yeah.
Like how often are rats in the wild going to have all these wires sticking out of their brains, and then they're running on a treadmill ball thing? Like experiments are always going to have a certain level of contrivedness, I guess.
But it's also how it's presented, right? Like if it's not presented that it's this behavior that is like
I see
so
what you
cool,
mean.
but maybe that we're using this to answer these further questions, like the probes, the ball running isn't going to be seen. Like we're not. It's almost like the title of the paper may have been misleading. It's like we're describing imagination in this one particular scenario, and the neuroscience behind this one scenario, and using this scenario to answer potential questions about this. Yeah, so Gojira, I think it's definitely it seems like it's a bigger conflict for my field than for like in Danny's field, where you're not getting your funding or your question, like you don't have to go find a T-Rex in the field, right? It's like you find what you find, and
yeah. Well, There actually is a story about that, about having to find a T-Rex in the field, where back in the late 1990s or early 2000s, Jack Horner, my old boss from Museum of the Rockies, got a bunch of funding, some of which came like through the Smithsonian. The Smithsonian really wanted a T-Rex to put on display there because they just had a cast. They didn't have a real Tyrannosaurus skeleton. And so Jack said that he would go find them one. And Jack and some of his students got a little distracted with Triceratops and just dug up like 100 Triceratops, which is really cool. And you could do a lot of really cool science with that. But the Smithsonian was kind of like tapping their foot, like waiting for a T-Rex. So Jack just gave them one that he had in collections that they dug up in the early 90s.
That's kind of cool, though.
The Wonko Rex. Yeah. But that's the closest thing I could think of like, you know, having to find a T-Rex, you know? Yeah.
Danny, I want to make sure I'm respectful of your time. So whenever you need to leave, like, please.
Ooh, I should make myself some dinner probably in the next, like, half an hour to 40 minutes, something like that.
Okay. I want to make, yeah. Like,
is that fair?
Yeah, no.
Yeah.
Sir, you could.
Okay.
Please, this is you're kind enough to come on to the stream and chat with us, like whatever.
Oh, this
you
is
want.
great. I've been looking forward to this all week. Yeah.
Should we move on to a next paper? Should we?
Yeah, let's do that.
Speed hit 2.
That sounds great.
Okay.
Let's do it.
So this insect one I thought was really cool. So there is this endosymbiotic bacteria called Wolbachia. And it's one of those things that's so pervasive in insects where if you discover a new insect and you do some kind of genetic sequencing, there's always going to be a guy in the room that says, "Did you test for Wolbachia?"
Huh. And that's a whole genus right there?
Yep.
Wolbachia?
Yeah.
So is there a different species for each new species of insect that you find it in?
There is not yet really the resolution for it. So you can't culture them outside of the host. And so that makes it difficult. Like even here they did some sequencing and they did mention like when they did the RNA sequencing on the Wolbachia and the fly together, you have to do like special like almost genetic detanglement to make sure that things are going in the correct bin almost. Like these are fly genes, these are the Wolbachia genes that are expressed. And so yeah, like it's even if you have like a behavioral paper, like you catch wild caught flies, and you're like, hey, we saw this really cool behavior. There's gonna be a guy in the room who's like, do they have Wolbachia? Because they're just like they're in everything. And what's really cool about it is, at least for this paper, it seems like they're involved in fertility, this bacteria. And it's never been demonstrated before. So, and the reason I like this paper too is that it kind of brings into the idea of trade-offs, which I think is a really neat evolutionary concept, right? Of like, we evolve this phenomena, phenotype, or what have you, but what are you losing now? Like sexual selection is really big, right? You grow this really long tail, but are you going to be more likely to be caught and eaten? Right?
Yeah.
Because at face value, like without any context, this looks awesome if you're a fly, right? Like, let me get infected by this endosymbiotic bacteria.
And then it increases your fecundity? Like, you're having more offspring after that?
Like, what's wrong with that? That's awesome. It turns out Wolbachia infections cause holes in their brains to form, which wasn't talked about in this paper.
Interesting.
And it like just wrecks their neurosystems. Like learning and memory just goes right out the door. Which is again like a very interesting trade-off. I'm sure there's a balancing act and dosing and everything like that. We had Wolbachia infected flies in the lab, and we were told like, do not let them out. Do not let them into the other fly vials, because if they get in there and poop into the fly vials that are uninfected, you'll infect your other lines. And then all your data is going to sound and look really, really different.
Interesting.
And so yeah.
What I'm curious about is, can you pronounce the bacteria Wolbachia?
Wolbachia, yeah.
Wolbachia. What part of the fly do they live in? I know they're endosymbionts, so they're inside, but where are they specifically? Are they in the guts? Are they in the brain? Are they in the everywhere?
So here's again like I think this is where your frustration might come in. They're not a lot of the researchers don't take that holistic approach of looking at, because the Wolbachia are pretty much present throughout each of these organs, right? From the gut to ovaries to brain, like they're going to be pretty much pervasive everywhere. But a lot of these studies are looking at them at the single organ level, and so you're not looking at it in the context of the rest of the body and what they're doing elsewhere. So like that's what this paper is really like, ignoring the other data from other tissues.
Right. They're compartmentalizing because, well, that's what they can manage that, at least when they're looking at one specific organ or something.
Well, I get you have to start somewhere. It's just it ends up frustrating me too. Is like, I would want it in like almost a bigger picture context. Even when they did sequencing in this study to look at RNA levels, and they like it's only in the ovaries, right? And it's like you have the tissue of the organisms, like you could dissect. It's just gonna be, I mean, it's the price, right? It's really what it comes down to. It's like, do you sequence the brains as well in addition to the ovaries, or you just do only the ovaries?
Right. Yeah, that makes sense. Yeah. And it's interesting that they just go everywhere. Yeah, I don't know. I guess I'm used to thinking of, like, in human beings, for instance, the majority of the cells in our body are not our own, right? They're of our gut flora, but those are, like, kind of restricted to the gastrointestinal tract, I think. Whereas these guys are just going everywhere throughout the fly's body?
Yeah. And what's cool is they're not just in fruit flies, but they're also in mosquitoes, which is where actually the whole point of this paper is kind of going, is that if you can infect a mosquito with Wolbachia that has some kind of genetic alteration that maybe makes them, like, because we've been genetically manipulated males so that get them to mate with females and they're sterile, and so then you're artificially lowering the mosquito population. Or if you mate, you can also cure mosquitoes of malaria. You can introduce a genetic factor so they can never get the malaria parasite. Imagine if you made that mutant mosquito that is now going to get rid of malaria, essentially, and made them much more fecund. Like you can almost artificially
so they could just outbreed. Yeah. Interesting.
And so like that's like the goal here. And what was the wildest experiment to me was this one. There's this mutation, this MEIP26, that when mutated, no Wolbachia, nothing with Wolbachia, just the fly has this mutant gene. Its ovaries, like in C, D, and E, the stem cells are just wrecked. They're not working as well. Their egg numbers are lower. Like the morphology of the ovary is just totally off. And then if you infect them, like if you do an infection of them, then all of a sudden they like regain that structure. And so the Wolbachia are able to correct a genetic mutation of the animal. And it's just like, that's just like this endosymbiote, right? It's wild to me.
That's pretty wild. Wow.
And it's not by modulating that gene, because that gene is removed. So they're changing other genetic factors to compensate for that, to fecundize, if that's a verb.
Yeah.
The original animal.
And I guess that makes sense, because that's going to benefit the bacteria as well, because the bacteria presumably will be spread through the offspring too, right?
Yeah, so they didn't do a good counter stain on these. There are stains where you can see, oh, here we go, actually. So this Propidium iodide is looking for genetic material, both RNA and DNA, so that's in red here. And they had some nice comparison where there's just an increased amount of red density in the ovaries that are infected versus the uninfected. So you're not making out what is the Wolbachia and what is fly, but you can just see like the increase in genetic material. And so that's like one way to, I guess, visualize that, yes, there's in fact more in here.
Interesting.
But, and also, I just really like the pictures of the ovaries. I think they're really pretty.
Yeah. No, that is pretty cool. And this is. So how do they color these? Is this just like because this is clearly not done by hand. How do they manage to color code that in a computer?
So, it's 3 different stains that you have to end up doing.
Oh,
So,
gotcha. Okay.
The
makes
PI is this propydium iodide. The VAS is vasopressin, and I think the orb is actually a gene called orb. I'll double check the figure legend. Yeah, and essentially, so the propydium iodide is just you can order it. Finger paint, Tommy. Tommy Clark. You can order. I can order propydium iodide to come to our house, and you just take a droplet of it, put it on a sample. It could be living cells, and it'll do this in red. And there's a red fluorophore, so that when you shine a particular wavelength of light on it, it excites it and it emits red light, and then your microscope sensor picks it up, and it's just whatever's glowing in that field is the propydium iodide. So if you like here in this panel J, that's just the orb signal.
Okay.
Here, and so you look at different filters, and so here you'd have 3 filters of different wavelengths of light, and so orb. They're probably false coloring stuff, right? This is what you see under the scope is this white, and then you can add, like in Photoshop, like, okay, I'm gonna make this white cyan, so when I make the merged image, it's visible to everyone.
That makes a lot of sense, yeah.
But like the orb and vasopressin are antibody stains, so essentially what you do is you purify the protein from an insect or honestly whatever you want. You take that purified protein and inject it into a bunny rabbit or a llama or a goat, and they have an immune response against it, right? Because it's a foreign, let's say, insect-specific protein. You then do, you take their blood and check and see the antibodies, and you isolate the antibodies for that particular molecule, and then you use that for this kind of stain. So if you put in rabbit antibodies onto a fruit fly sample, if the rabbit antibodies are recognizing this very specific protein that you injected into the rabbit, the antibodies will only bind to that one thing. And then you can attach a glowing molecule to the end of it called a fluorophore.
That's really clever.
Yeah, and it lights up. And so it's the hard part is actually just isolating and making a ton of the original
protein. Sounds like
a
tremendous amount of work. Yeah.
Yeah, it's because you have to synthesize the protein. Like, you need that genetic sequence of what the protein is. You put it into bacterial cells that produce a ton of it, and then then essentially then when you have a ton of that protein, you send it off to a company, and they do maybe even 2 rounds of injections. So like, especially with mammals, you can immune prime an animal, and you inject a second round, and then they have even more special antibodies to attack that particular foreign substance, and then you're sent a vial, like a 50 milliliter tube of of like rabbit blood, basically. Well, it's like purified out a little bit, but that's essentially what you're getting. And you might be like, well, is that really enough for, like, how much is that enough for? You dilute that down, and that could be enough for 20 years.
Yeah.
And so it's each one of these you can do that with. Now, these vasopressin and orb are so popular. There's actually a database called now the name's escaping, but you can go onto their website and order it. Hybridoma. It's the Hybridoma Bank, and you just search for the organism and the protein, and they're like, if they have it in stock, you order it and they send it, and then you can run your test. You don't have to wait like a couple of months to actually build a custom one.
Right. That's really cool. Wow.
Right? It's
pretty neat to hear about. Like, I've zero idea about any of this stuff. It's this whole new world, and that's really, really cool.
Yeah, and that's again one of the reasons I like the collab stream is that we can talk about these kinds of things. You know, it's like for me too. I'm like, Danny, I have no idea what this is, and presumably other people in our 2 audiences will have similar questions as well, or maybe we'll spawn a question off it that's, similar from our naivete. But yeah, I just thought it was a really cool paper. It had really pretty pictures. I thought the summary figure was a mess. So this was trying to tie together what genes were actually acting on what, and
Okay.
this they were pulling from other studies as well, and so it just kind of is an aesthetically also like gobbledygook mess. But they're trying to say like, this kind of thing inhibits this gene, which inhibits this gene, and then there's their weird. They could have been done a much better summary figure. It's not entirely clear what
Greek
exactly.
to me, yeah.
Yeah, they're trying to show off, and I think like this is part of the issue, right? Is that they pull in citations from past studies. This is at like the genetic level of like a transcription factor turning on a particular gene, and then this is supposed to be an ovary interacting, so it's showing so many different things that it was a little bit lost to me. But they did find like what the Wolbachia was acting on in terms of genetic factors, which was kind of neat. They pulled out 6 possible genetic targets that the Wolbachia infection was messing with when you introduced that mutant, which was, again, it's that an endosymbiote manipulating a host genome, and then that resulting in changes in physiology. And we've got a raid from Malia Illa. Malia, welcome on in. How was your stream? Tell us all the things, what you're up to. What were you making and crafting? Tell us all the things, Malia. Welcome in. My name is Balint. I'm 1/3 of SciAnts streams. And we've got a special guest today, Professor Danny Anduza of Paleontologizing. The single greatest paleontologist, not just on Twitch, but the entire internet. And we're chatting fossil science and genetics. Raiders, welcome the heck in. I hope y'all had a great stream. Let us know how it was. And feel free to ask us any and all questions about the science we're chatting about tonight. Welcome the heck in, y'all. Made some 3D gingerbread houses and some little gingerbread house cookies, too.
Very nice.
So, Malia,
Cool.
edible, 3D. Oh, from polymer clay. Okay. I already answered my. I was like, wait a minute. What kind of deliciousness?
Nice.
No, we probably shouldn't. Eat. Malia, thank you for the rate. I love polymer clay. We've got a bunch of friendos that do polymer clay stuff and makers and crafters. So I'm excited to check you out. If you have anything you want to share with us, feel free to drop it in the chat, and we'll check it out in just a little bit. Again, welcome on in. And Kixie and Spectrum 13, welcome in as well. How are y'all doing today?
Hang on, I can't hear.
Not edible. Yeah. Glad. Please do not eat. Malia, we've had soap makers come in as well who always tell us to not eat the soap. It's a commonplace theme. But I was just excited. Gingerbread. I love gingerbread.
Earbud is dying here. I'm trying to get it. Okay, good. I think I got it.
Nice.
Cool. Move on to earbud number 2.
All right. With that, I know you've got to make dinner soon. Do we jump to the third paper, or is there anything you wanted to point out, or fourth paper? Do you want to point out anything from this?
It's just cool stuff. I don't. It's neat to see you get excited about this. And yeah, to like get an inside scoop on this kind of thing is always really cool. So
yeah, I really like the imaging, and there was a ton of sequencing, and this is like a lot of this is this kind of figure data. I don't really enjoy these panels very much because they just there's a lot of weeds here, but you can look at the different levels, and for everyone's interested, we'll post on the Discord the papers and that we chatted about tonight. Actually, I think they're already posted, but you can find the variances in the levels and the gene expression. What's always frustrating is the axes. So I'm sure you can see this too, Danny, is that the y-axis in every single one of these is different. And so like my human eye, I just like to look at across and see like what the differences are. But because the axes are always different and there's nothing pointing out that these are different, it's just frustrating because you're like,
you'd hope they'd be consistent. That's the point of it. Yeah.
Even within one of these boxes, right? Like, 8. And then it's just so it's very hard to have that cross comparative approach to it. But they did the sequencing. They have these insane interactome maps, and then the proposed genetic regulation, and then with a bunch of the imaging is really the big thing for me on this one. Is like the convincing
cool stuff.
Yeah.
And now that I actually know the process like that you use to create those colored images, it's so much more impressive now that you described that. So thank you for that.
It's so one fun limiting fact of these. So the way to get it to be the more long lasting for the antibody, and this is getting into the weeds of things, but you don't fuse that fluorescent probe to your antibody to start with. So if the normal way you have it is you have those antibodies, and if you put it onto your sample, nothing's going to glow because there's no fluorophore. And you can fuse it to your primary antibody, but that means that you're always stuck using that one color. Right? And you could imagine a situation where you want to stain two different things, but if they're both the same color, like you can't tell them apart. So what you do is you have a primary antibody, so that's your rabbit antibody recognizing your protein of interest. And then you put on what it's called a secondary antibody. And so your secondary antibody is very generic, and it recognizes, let's say, rabbit. And so what's cool about that system is, in a fruit fly sample, the only thing that's rabbit is the antibody that you've put in.
Gotcha.
And so it's only going to bind to that one thing. And then that's what fluoresces. It's that secondary antibody.
That's pretty cool.
And so that way, you can just hook up with as many colors as you want.
Neat. Very neat.
Yeah.
Yeah.
And Melly, thank you so very much for the raid. Have a lovely sleep, and we'll see you next time.
Cool. Should we move on to the final paper? This was one that I almost kind of felt bad for choosing this one. I wanted to choose two papers from this week, and I should have also extended that into last week too, because I forgot it was Thanksgiving week, and Did you have a good Thanksgiving, by the way? I didn't even ask. Where are my manners?
No, we both just got into the science. Let's go.
Yeah.
It was Baby Alona. Turns out loves turkey. She loves turkey.
Oh wow.
So she had this big slice of turkey breast, like the thin slice, but we were starting to give her the pieces, and she ate an entire piece, just like kept going and going and going and like it was like, little one, you gotta cool it here. But it was good. We went over to Lita's mom's and had a nice meal together, and
very nice.
It was a good relaxing. And then we had a community game night stream that night. So for anyone who didn't have anywhere to go, or if they were sick of their families, we did a game night, and that was a good time. What about you?
Very nice. Yeah, I went up to my mom's place. So my mom and her sisters and some other family members, my brother and his wife, and it was nice. Yeah. My mom just recently moved up near Sacramento, and so I helped her move into her new place probably about a month ago, and she's settling in pretty well, and it was nice. Yeah, I also brought my streaming laptop and stuff up there to try and stream, and there just was no time for it. So didn't get to do any streams for 2 days.
We've had crossover streams with your mom before, who's been on, right, with the laptop, if I'm not mistaken, right?
Yeah. This was a couple years ago. Yeah. My mom and her sister, my Auntie Kathy.
Yeah. Because we've been. I just had my sub-anniversary with you a few days ago. It's been 26 plus months, right? So nice. It's I have all these cool memories of your streams about the museum tours and the field research, and even the family members, or with Ios and Lordy, the cocktail hours that y'all have done, the barbecue on their patio, all these cool events, and it's always been it's always a lot of fun to look back and man, it's
it warms my heart to hear that. Yeah. Cool. I know we met for the first time, you and me, in person at TwitchCon, but I feel like I've known you for a lot longer. So
yeah, that's I think that's
Funny how that works.
the cool part of TwitchCon too is there's so many people that we met for the first time, but it didn't feel like a meeting. Like there was no like this awkward introduction. It was just
it felt like a reunion or something.
Yeah. Like, I haven't seen you in a while. Like it's so good to see you. It's the nice part of Twitch too is that you can kind of select your friends and you almost pre-screen them based on the stream or the interactions you have with them, and then when you meet them, you're like, yeah, we're already friends. Like, let's go. It's all good.
Yep. Yeah. All right. Should we get into this last paper? So I feel kind of bad about picking this one because it's not the most exciting paper, but I thought it was interesting to pick one that's like a rebuttal to a previous paper. So this is basically like rebutting my old crew chief, Denver Fowler, published this paper about anagenesis in tyrannosaurs. So are you familiar with these anagenesis versus cladogenesis? Are these terms that you use in your field?
So we don't use them regularly in the field. I can't remember any paper where we've talked about that. Or like that's written out as like a phenomena. Like any evolutionary molecular studies are on the order of like bacterial evolution or maybe some like phylogenetics at best, but even then like these like the anagenesis and cladogenesis aren't commonplace. And so I think I understand anagenesis as like, and correct me if I'm wrong, it says like as you're evolving over time, how the animal's changing. Right? Is that a
that almost sounds more like heterochrony. Anagenesis is basically just evolution without any splitting that happens.
Okay.
So it's like linear evolution, non-branching evolution. So rather than like things splitting into different populations and then different species, it's just one population continuously changing is anagenesis.
Yep. That's right. Yeah, that's what I was trying to go.
Versus cladogenesis, which is like new clades forming. So like you've got a split. There's no longer genes being passed back and forth. These populations split and eventually they become different species. Yeah. And so so with Denver's paper, they found these Daspletosaurus fossils and they said, holy cow, they look like a new species of Daspletosaurus. But Denver said, like, hold on, are these just like actually a new species and that like there was a split? Or is it just that these are a little bit older than these other Daspletosaurus that have been found before? And it's just like one population slowly changing its morphology over time. He thought that was the case. And this is like an ongoing, maybe growing controversy in dinosaur paleontology. Is like, how common is anagenesis? Because there's just, I think by default, paleontologists are used to thinking of, if you've got a fossil that looks different from another one, it's got to be a cladogenetic event. It has to have been, they split at some point. Now they're different lineages. But...
Oh my goodness, we've got a giant raid from Mop Garden. Raiders. Welcome the heck in. Mop Garden, how was your stream? Tell me all the things. Oh my goodness. We met Mop Garden very recently. Amazing gaming streamer extraordinaire. Y'all were playing the Lies of P.
Nice.
Oh my god. No sorries, Mop Garden. How dare Mop Garden. Guys, go check out our friend Mop Garden. How was Lies of P? Joshua Sweet, thank you for that tier 1 gifts up to Mop Garden. How was Lies of P? It is a Souls-based game that is extremely difficult. I hope it was fun. Y'all had a good time. Bill Buttlicker69, amazing name. Welcome on in. Bub Rub for real. Hello. Dwin, the lovely Mop Garden, of course. The OG Mana. Hello. It was great fun. Yeah, if there's anything you want to share, Mop Garden, please let us know. We'd love to show it off with the party people. For the Raiders coming on in. Raiders! With another raid. We got double raided. B4UX, welcome in as well. For all the Raiders, I'm Balence, so I'm 1/3 of SciAnts.
Is it Bauxite? I think that's the name. Is it Bauxite?
You're right.
Yeah.
No, the lead gets me. The lead, I cannot. I just say B4UX, the box.
Yeah, nice.
Yeah. Yep. Bauxite is an amazing programmer extraordinaire. Again,
Oh, very cool.
for y'all raiders coming in, y'all have entered a science stream. I'm part of SciAnts. I'm an ant geneticist. My name is Belent. I'm 1/3 of the team. The other two are my lovely wife Lita and our daughter baby Alona. And we have a special guest on today, Professor Danny Anduza of Paleontologizing. Please go drop him a follow. We'll give him a fancy shout out in a moment. His name is also in the bottom of the screen description that you can click on and easily drop him a follow as well. We're both scientists in our respective fields. We do field work, we do work in the lab, and we are here on Twitch to bring you closer to the science than ever before. To ask questions and, yeah, question us, whatever we can do to get you excited about science. That's what we're here for. Tonight we're talking about different scientific papers from our own unique field. So Danny is talking about, he's an expert in dinosaur science, and I'm an expert in genetics, and so we're going back and forth on different papers in our fields and what our views of them, and asking each other questions and having y'all here too and asking questions as well. Does it make you feel typical? Oh, thank y'all for the follows, by the way. Mop says, how does the typical human evolution diagram make you feel? Do you mean, Mop, the coming out of the water and the increase in height and...
Oh yeah, the descent of man.
Yeah.
Classic image. Or are we talking about an actual phylogeny here, like an evolutionary family tree?
From the monkey. No, from the monkey. Look, from the descent of man.
Oh yeah. It doesn't work that way. It's much more complicated, and yeah.
I think Mop, it's a little frustrating when trying to talking about why. How evolution might work in the study of different organisms and how that's being utilized. I think that's always a big difficulty in the field, especially because the image is so pervasive, right? It's everywhere. Mm-hmm. And it makes it kind of difficult to chat about and how exactly those things work. But it's fun.
Yeah,
it's a fun talking point. Guys, also go follow our other raider. Joshua Sweet, thank you for the 100 biddies. Go check out our other raider as well. I cannot remember B4's names in the lead, but B4UX, our amazing game dev friend extraordinaire. Again, y'all, welcome on in. Please feel free to ask any and all science questions, and more about our backgrounds. We had a panel at TwitchCon advocating for science communication here on Twitch. We are, trying to lead the charge on making educational content and bring you ever closer to the sciences here. And Danny actually does streams from the field where he's digging up dinosaurs. Please go follow him. Please follow Danny. He's so kind to bring, come on the stream and hang with us. So
yeah. Oh, it's my pleasure, Belinda. This is great.
Bauxite, I'm big gamey. I wish I could remember that, but 5 minutes from now, I'm just gonna go right back to B4UX. The one we're talking about now, for y'all coming on in, is this paper we were talking about anagenesis and cladogenesis, in particular with Tyrannosaurus lineages. Earlier tonight we talked about oogenesis, so egg production in fruit flies, and longevity, and so how old parents give birth to different quality offspring and embryos in fruit flies. We also talked about a really cool study in Troodon looking at teeth structure and signaling molecules for isotope readings to suggest what their diets may have been. And so we've been hitting the gambit of interesting science tonight. So please, if you have any questions, feel free to ask us. That's why we're here. Kennedy actually has a question. Thank you, Zoom Boy, for that follow. Danny, you talked about linear evolution versus branching your stream. What do we know about that in ants? So Kennedy, we know, so as Danny says, birds are dinosaurs, right? The equivalent of that in this line of work in insects and ants would be ants are wasps, right? So the evolutionary originator of
This is a dinosaur. This birdie.
Indeed, Tom.
Birdie dinosaur. Dinosaur birdie. Birdie dinosaur. Dinosaur birdie.
So your ants, modern day ants, are descended from wasps that had some level of social interaction, but very, very little, and no real division of labor in terms of reproductive versus worker, and you have that now in your modern day ants and bees. Now, where those branched, it's so difficult to tell, because we think like one of the earliest ants that we have a sample of is, collected from amber resin, and that's the hell ant. And looking at Spectrum, thank you for those biddies. Looking at the hell ant. Thank you, Jaden. Looking at the hell ant, we have no idea where they stopped in terms of evolution, because there's such a gap in the fossil record.
I like your pants around your pants.
Thank you, Marissa. Pants around your pants, indeed. There's such a gap in the fossil record that we don't know at what point that lineage continued and what point it split. We do know all those vertical jawed ants went extinct, presumably because of over-specialization, but where that bifurcation point was, where you had ants with these kinds of horizontal mandibles versus the vertical, and what point there was the movement of those, we just don't have it yet. There's such a big gap in those fossil records. But great. I wish I could tell you, Kennedy, but they're just not there. I mean, I know, Danny, you probably have the same issue as well with a lot of the samples, where it's like, I wish, like we with the wrist bones, we were talking about the Spinosaurids not having that, and that seems like a fundamental behavior. But what do you do with that?
We call these ghost lineages in paleontology, where it's like, we've got something at the bottom, and then we've got modern creatures today, and we don't know what the things in between look like. So we have a lot of that kind of thing for sure.
Yeah. And guys, go hit that follow button on Professor Anduza right now. At the top of chat, you should have that heart button if you're not following him yet. Go hit that heart button, chat. Sorry, go ahead.
Yeah, go ahead.
Coffee Rocks was wondering if the difference between the evolution of humans versus how we branched off from other primates, that's more what we should be looking at versus just like coming from a monkey. It's just like the branching.
Oh yeah. There's I don't know. It's not like a continuous line for humans. There's like all kinds of different species that branched off and did their own interesting things, and which kind of makes some sense, because we were like pretty specialized creatures, I guess. Well, I don't know. Maybe that's not fair to say. But one thing that's kind of interesting is that, like, it seems like the larger an animal is, the more difficult it is to make it speciate. Like, in order to, like, separate populations so they can no longer pass genes back and forth. It's like, think about how many species of elephant there are in the entire continent of Africa today. 2. But, like, how many species of moles or voles maybe are there? And you can have a lot more voles in a given environment than you can have elephants. And so they also have faster generational time, so like they're producing offspring faster and stuff like that. So they can like evolve more quickly. It's easier to get them to separate them into different populations where they become their own species. It's a lot harder to do that with elephants. Elephants just go wherever they want. It's hard to actually make them be geographically
one is
isolated.
not tethered by earthly limitations. What does that mean?
Well,
so
come.
It's easier to like observe cladogenesis, and it's easier to make that happen in small animals than it is in large animals. And so my old crew chief, when he published the paper that this one is a rebuttal to, the idea is basically like, these are large-bodied animals. They're very mobile. They're very adaptable. It's hard to get them to like speciate, and so the changes that we see in these are probably anagenetic. These are not like branching events. This is one continuous population evolving over time. But a lot of paleontologists are like really hesitant to accept that idea, because it's just easier to name things as different species and say like, oh yeah, well there's a splitting event somewhere in the past. We'll find it eventually, maybe. So
I like that.
It kind of gets into like a philosophical difference,
Yeah, like how you were talking about the sizes, right? Of like even like your modern day insects, it's easier to cause there to be like genetic changes, the short generation time, and presumably the dinosaurs that are, like you said, are bigger and bigger, would be harder to have that happen.
Yeah.
Octavius, thank you for that gift sub. to Mom Boss
Nice.
Lady. And Mom Boss Lady, thank you for following Professor Anduza of Paleontologizing.
Yeah, and like when you're a large mobile animal, it's like it's harder to put geographic boundaries in your way. With insects, like maybe they might be separated by like a river or something like that, and suddenly they become two different species. But if you can cross that river just by swimming across it and then spread your genes elsewhere, you know, it's going to be a lot more difficult to actually get you to speciate.
Yeah.
Yeah.
And we had a question about, are there any especially basal species of ants today? So Smay, there are ants I've had the pleasure of working with where the workers can actually become queens and then revert, and so that level of plasticity we think is the more ancestral state, similar to how there are wasps even today that can make a pseudo colony where they underfeed their babies and the babies come out smaller, and so there's a size hierarchy. So there's a colony, but it's not because there's a reproductive division of labor because of the genetics. It's rather because it's like, well, you're the little guy and you can't reproduce and the big one does, and so it's kind of harkens back to that kind of time, which eventually the next step was there was a queen and workers, but if the queen dies, a worker can easily replace her, and then you get to your modern day colony setups. So Danny, with this paper, how do you feel? You said you had thoughts about how you felt about the findings.
I mean, they use phylogenetic analysis in this in order to like make a case that like, oh, it can't be anagenesis here. This has to be cladogenesis, like branching evolution. But I don't know. Phylogenetic analysis has kind of like a weak spot kind of built into it, and it can't really show ancestor-descendant relationships. When you're building a cladogram like this, it can only show that that things are related, like sister taxa. It can't really show that ancestor-descendant relationship if one was the ancestor and one was the descendant. It just won't come out in the analysis. And there's various ways that you can tweak it to try and make that happen, but I don't know. I almost feel like it's kind of using the wrong tools. Yeah, and that being said, I want to also clarify here. I didn't get to read the whole paper. I think because it was this one was paywalled, wasn't it?
I'm not paywalled on this, so I can send you the paper.
Oh, okay, never mind. Oh, so I was thinking of a different paper earlier this week. Yeah, I didn't have time to read the whole thing.
I hate paywalls too. There's so many.
Yeah.
Naive, silly question. How do they build these phylogenetic trees with any
Since we don't have genes,
right?
Yeah.
With certainty. Where
it's a lot less objective, I think, than coding characters genotypically. So we have to create morphological characters and code them basically at our discretion. So it's say the lacrimal crest on a theropod dinosaur, and it is twice as tall as it is broad or something like that, and we code that as a 1. And then if it's not like that, it codes as a 0. But they're subjective, So and there are certain things that are not actually diagnostic. So maybe there's a lot of individual variation in this, and or maybe it's something that changes a lot through the animal's lifetime. You can have bad characters like that just mess up your entire analysis. And likewise, if you're taking an individual dinosaur specimen and putting it into the analysis, and that one is a juvenile, that'll screw up your whole analysis as well, because juvenile animals, they tend to look more like the primitive character state, so it'll look more like the ancestor, and so that will usually come out as very basal on the tree, and it's oh wow, we've got this very small animal that it looks really, really primitive. It's well, it's because it's a juvenile. This happens again and again and again in dinosaur paleontology, and it's really frustrating.
Yeah, I mean,
to see people make the same mistakes.
You were telling us too, the misnomers of different species, just because, and it's not a different species, but rather it's just a juvenile, and so it's
yeah.
Yeah, that phylogenetic tree does seem like it could have a lot of issues.
Yeah. Part of what they're doing here is they're saying that that Daspletosaurus and Tyrannosaurus don't belong to the same lineage. I think if we go back to the cladogram like you were showing there earlier, I think they're saying that they're two different lineages.
Yeah.
Yeah. And it's interesting, but like this is all going to come out differently depending on how you code these different characters. And I didn't actually look into see if they used the same exact data matrix as Warshaw et al., which I think was a paper from last year. That was my crew chief was an author on that paper. I'm not sure how to pronounce the first name. Elias or Elias? Elias Warshaw, he's an undergrad, and he was first author on this big paper describing a new species of Tyrannosaur.
Nice.
Which is really exciting that, like, he got to do that. He's going to be, I think, a rising star in dinosaur paleontology. But, anyway, yeah. I don't know if this rebuttal paper used the same character matrix and everything as that one did. They put some other specimens into the analysis, and that kind of shook out it gave a different aspect to the tree. Like the dinosaurs came out in different places on that family tree because they put new dinosaurs into the analysis. But like so much of this is totally dependent on how you code those characters. There's, I think, a real sense of false objectivity in a lot of this stuff, where like the character states are not objective.
Hi, Hugen. Hugen, thank you very much for the raid. Hugen, how was your stream? Tell us all the things. Hugen, you streamed twice today. You also raided Danny today. Thank you, Hugen, for the raid. How was the stream? So it seems like these matrices can be very different based on the features that you're doing, right?
If you code the characters differently, you'll get completely different results.
If
and like the number of
the
different specimens that you put into it, like if you put a juvenile into a tree like this, like it can shake things up also, and it might come out as super basal. And there have been times when like someone has coded a juvenile Tyrannosaurus as a different species, and it's like, oh wow, we've got this really primitive Tyrannosaur running around in the same environment, and it's like it's literally the same species, but it comes out way at the base of the family tree.
And if they're trying to compare,
this kind of thing happens a lot.
Other data, right, from a different paper, but they're using different features, won't that inherently bias the results of this new one?
Sometimes you can use the same like data matrix.
Yeah.
And like, so you're able to control for some of that, but yeah, I don't know. I don't really know enough about some of the methodology to say whether or not you would get different results just by using a different program. Yeah, it's all parsimony based, but like different programs will have different ways of determining parsimony, as I understand it.
Yeah.
And I don't know how much that actually affects your result. Maybe it doesn't at all. It'd be nice to hear that it doesn't at all, but I'm not sure.
If it's anything like in our fields, different programs will drastically affect the data outcome.
Hugen says like bootstrapping and like all kinds of stuff like that too. Yeah.
Hugen says, I'm happy to announce that my stream works on all fronts. I'm back to streaming. Excellent. Congratulations, Hugen.
Very nice.
Glad to hear it's going well. You did everything from did you play a game tonight, Hugen? Because I know earlier today you're doing a walk outside, so that's awesome. I'm excited to hear you getting back into the swing of things. Excellent. Thank you again for the raid, Hugen. Yeah, so this is I thought this was a really cool paper, and even though it was like just the approach of it, not necessarily what the finding is, like, because I'm not one to.
Well, this is kind of your introduction to dinosaur phylogenetics here, I guess.
Yeah.
Like dinosaur cladistics, yeah.
But it's, I guess, like getting the answer of can I judge at value what this is, like if I agree with that I don't feel like. If it is...
You don't have a horse in this fight,
Right. I mean, I like the hypothesis of anagenesis. To me, that, given modern examples of sizes, like, I like that idea, but I also recognize it's almost harder to...
Yeah. Well, that's the thing. So first off, like, anagenesis this is not like the consensus view among dinosaur paleontologists right now. Like, this is kind of an outside idea.
Yeah.
And, like, by putting forth this idea that, like, yes, anagenesis is really common in dinosaur lineages, like, our work is cut out for us, because that's not the consensus right now. I think it makes a lot of sense, and I think it, in some ways, like, it comes down to, what is your default hypothesis? What's the null hypothesis in this situation? Is it that you've got branching evolution? Is cladogenesis the null hypothesis? Or should anagenesis be the null hypothesis? And, like, it kind of comes down to, like, a difference in philosophy. But the thing is, if you have very few specimens, cladogenesis is always going to be the default assumption, because you're not going to be able to see those smooth gradations between one ancestral species and its descendant. They're just going to look like two different things. And so I think there's kind of a real bias, an unfortunate bias, toward cladogenetic thinking in dinosaur paleontology. But I'm in the minority there, you know? Other paleontologists go, well, of course, you know, it's going to be cladogenesis, because there's this assumption that, like, evolution only really happens when you have a split between species, that, like, individual species themselves don't really evolve much. It's this whole, like, punctuated equilibrium idea. Anyway, I'm getting way into the weeds here, but it kind of gets into, like, fundamental differences in the way that we think about evolution among different paleontologists.
It's just it's such a cool concept to be able to track, but it also seems like it would be extremely difficult to figure out when there's an actual split point versus just a singular population moving forward, because that's in a way a split point, but not really the same kind of split point as a cladogenesis split point. So that part was super cool to me, just on the, like, the theory side of things. Again, like, in terms of what the right answer is here, I'm not well versed enough to say. Again, I like the anagenesis hypothesis because that feels more parsimonious to me, and so that chat is just like, what's the more simple explanation is more likely what's gonna be the thing that evolved and happened versus constantly producing new species. It kind of makes sense to have one population just changing over time.
But other paleontologists would be like, well, you know, what do you mean, like, one population changing over time without splitting? Like, of course they're going to split, and you're going to have new populations form, and it's like, what actually is the most parsimonious answer? Like, we've got differences of opinion in the paleontological community about this. So yeah.
Harissa's got a cool story for you, Danny. We're just discussing to my daughter. We're discussing how long I've been following paleontologizing. So she was 9 when I first started watching him, and she's 12 now. She goes by Ash on your stream.
Oh, that is awesome! Holy cow!
It's kind of cool to think about how your kids are growing up with Danny in a way. Like, I know, Munchkin knows your voice. Like, like, she perks up and watches you and Irish John and Thug Shell's voices. She recognizes and perks up and watches. So I'm like, she's gonna know, you know, freestyle rapping, coding, and dinosaur science. I'm like, that's a well-rounded child.
It's a good combination, yeah.
But yeah, I know you gotta go make dinner. I don't want to keep you too late. Any other big picture things we want to take away from this particular study?
I think it's interesting. We don't actually have this all that often where there's, like, a direct rebuttal paper in dinosaur paleontology. Although, shoot, like we were talking about earlier with, like, different charismatic groups, Tyrannosaurs, very charismatic dinosaur group. You might actually have some of the most direct rebuttal papers in the study of Tyrannosaurs, just because lots of people are interested in tyrannosaurs because they're the last of the big meat-eating dinosaurs, and they're among the biggest of the meat-eating dinosaurs. And yeah, so it'll be interesting to see like what the general consensus is that shakes out about this in the future. But I think it's probably going to be like decades before we can really say any of this stuff with any certainty. I've got my own hunches, and other paleontologists have theirs, and it's just exciting that like we've got active debate going on about this.
It's also interesting to see a paper like this. I haven't seen a paper like this in our field where there's this kind of intellectual exchange happening. Because this is like an actual paper. Like there's data here. There is like experiments, right? Like the most that I see is there will be a commentary piece released where the it's not a data set, it's just like from a differing side a scientist writes like a rebuttal, but there's no experimentation.
So just trying to give a different perspective on it or something like that, rather than like actually presenting new data.
Yeah, and almost like this kind of study would be trying to push for a retraction of the other one.
Oh,
like it doesn't come across as like a dialogue like this. Like this didn't feel aggressive. This just felt like a different way to analyze the data.
That's funny because it did feel aggressive to me a little bit.
That's cool.
Yeah. But again, that's different perspectives from different fields. Like this feels like a shot across the bow because we don't have like retractions of papers in paleontology are extraordinarily rare. I can think of like 2 from the past 20 years. Maybe a few more. Fewer than 5. But anyway, it's like this is interesting stuff, and like this is the sort of thing that like people will argue about at meetings vehemently. And it's interesting. I'm excited to see how this sort of thing shakes out. Because I've got my own opinions about this, and I'm actually interested in publishing on anagenesis in the future. But, I could very well be completely wrong about this, and that's kind of exciting. Like, it's cool that, like, we as scientists, we can take joy in being wrong sometimes, and being proven wrong. Because either outcome, whether I'm right or wrong, I'm going to learn something new, and it's going to be really cool.
Yeah, I mean, those are the best scientific questions, right? Like we were talking about earlier tonight. You get a cool result no matter what answer you get. And
definitely.
So I'm subscribing to this idea of anagenesis. I'm like, I think this makes total sense and it's cool. But if you told me that it's really like it's constant clating that happens, I think that's really cool too, and it speaks.
We just have lots and lots of different species of tyrannosaur running around at the same time. Like, that's also really cool.
Yeah, and like why that's like speaks a lot of the natural history of the animals and what drove them to these, like what drove them to evolve so many times, where these clades split versus just like a general trend towards like you can population like so many questions come into play on both.
Definitely. We could talk about like the stability of these ecosystems in the late Cretaceous, and like are conditions changing all the time, and like that would cause speciation, or is it like relatively stable, and a population will just evolve in a linear fashion. Like there are larger questions that work their way into this too, it's really cool how integrative it can all be.
Yeah. Let's get one more question because I know you have to go make dinner, my friend.
Sure.
Bub was wondering. I apologize if my question is too rudimentary. No sorries, Bub. How does a paleontologist determine where to first start digging in a new area for a fossil? Is there a way to predict where you would start digging a fossil? Is it more like, hey, someone found this thing that looks like a bone, let's look there?
A big part of it is that, and I've actually got a question for Bub Rub for real as well. Bub Rub, is your name a reference to that viral video from Oakland with the whistle tips, Bub Rub and Lil Sis? Because if it is, good on you. Small world. But let's see. If we're looking so many like different it isn't a reference to that? Yeah, the whistles go woo. Yes, indeed. Classic Classic East Bay area video bullet. We'll have to watch it sometime. That's like down the street from where I live.
Oh, that is a really small world. Nice.
Yeah, seriously, yeah. Yeah, so many like important dinosaur finds are just made by people out, not by paleontologists, but just by like either amateur fossil collectors, people who are just enthusiastic about fossils, or by like people out walking their dog out in the Badlands, or bird watching, or hiking, or stuff like that. So many like really important finds are just stumbled across by regular people. But if I'm out on a crew and I am like prospecting an area for dinosaur fossils, we will go and pinpoint certain areas because we've got rocks of the right age and the right type exposed on the surface. So it's not like we just go dig a random pit somewhere and hope to find dinosaur fossils. We look at geologic maps to figure out where you've got rocks from the age of dinosaurs. Maybe like we're looking at the end of the age of dinosaurs. Say I want to find a Tyrannosaurus. We'd find rocks from, say, the Hell Creek or the Lance Formation. And you find those in places like Montana and Wyoming and the Dakotas. And then you go out there and you find rock, like where those rocks are exposed on the surface. And you go out and you look. You use the old Mark I eyeball to just scan the ground for bits of bone that are eroding out of the surface. And then that's really the most difficult part of the job, is being able to look at fragments of bone on the surface and determine, A, are they bone in the first place? B, what sort of a bone is it? From what part of the animal? From what kind of animal? And then you have to make a determination, like, is this worth digging in right here to see if there's more there, or should you continue on walking? Because you could spend all day just digging up a garbage rib, and then you only have so many days in the field season, and you can come back completely empty-handed. So that's really like the most difficult thing, is like being able to understand what is this bone that you're looking at and what is its significance. Because you might be looking at an animal that's the size of a bus, and it's got 200 bones, all of which can fragment in a zillion different ways, and they can weather in a zillion different ways. And so like being able to identify a chunk of a bone and say, oh yeah, so this is part of a, Triceratops, like cervical vertebra, one of the neck bones. And then, you might look at the surface texture and the interior texture if it's broken to figure out it's from Triceratops. There's like you need an encyclopedia of knowledge, basically, in order to be able to identify something based on a little fragment. And the people who've been doing field work for decades are really, really good at that. I've got so much left to learn and so much more experience to gain before I'll ever be at that level, but there are people who do that, and it's pretty extraordinary.
Which is insane to me, because I know the moment Lita and I started watching you, we're like, this guy knows his stuff so freaking well, because like questions you get, you're always just like boom, 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 like you just, and it's so, that's, the process seems very wild to me. Also the fact that you aren't already like this, totally like knowing all, it's just like, I don't know. I just always assume
oh my goodness.
You have all the expertise.
I think I just speak with a certain degree of confidence, and that it counts for a lot.
No, you back it up. It's not just the words. Yeah, that's Sparky's follow-up was, if you find bone, like if a regular average person just finds bone sticking up, do we call someone?
Yeah.
Like, is there someone you can call?
Call your local museum. Yeah. So what you should really do is, don't try and dig it up, and don't like carry it off to a museum or something like that, because you might destroy a bunch of really important data in the process. But take some good photos of it in the ground, and then take a GPS reading, and then email that to a museum. See if you can find a paleontology museum nearby, or failing that, like your local ranger station, or if you're on like state land or federal land or something, like find out who administers that land, and send them that email. And the important thing is that you notify somebody who can go out and dig it up and actually collect all the data with it, because oftentimes the surrounding data is even more important than the fossil bone itself. If it's outside of context, it might as well just be a doorstop or a paperweight. You know, it's that data around it, exactly where it came from in the rock layer, that's extraordinarily important. And yeah, if it turns out to be a new species of dinosaur, you could have a new species of dinosaur named after you. It's happened a lot in the past, like where just some regular member of the public stumbles upon a new dinosaur and then, wow, they've got a degree of immortality now because there's dinosaurs named after them. It's pretty cool.
That is really cool. And most phones nowadays, if you have location services on, you can take the picture and actually swipe up and it'll give you the GPS coordinates for it. So you don't even need like a specialized piece of equipment to do it. Mm-hmm. That's how we actually tracked the ants when we were doing field collections. We looked at the photos from the previous years and we're like, okay, where was it? All right, let's start there.
Nice. That's really cool. Yeah.
Well, Danny, I know you have to go make dinner. I don't want to keep you too late. Thank you so very much for coming on with us tonight.
It's been a joy.
That was like a lot of fun. A lot of variety tonight too.
Yeah, for sure. Yeah.
I think
let's do this again next week.
Are you okay doing it next week? I know you're moving.
Yeah, next Thursday I might be busy, but let's try for the Wednesday afterward, maybe, or maybe the Thursday afterward. We'll figure it out.
Okay.
Yeah.
Sounds good. So y'all, we will continue this crossover stream as long as Danny's okay putting up with me. We'll continue doing this. Thank you so very much, my friend, for your time, your brilliance, your streaming, advocacy for science communication. You're, a pillar that we all look up to in terms of science communicators and trying to make a difference.
I could say the exact same thing about you, Boleyn. Thank you. Yeah, have a great rest of your stream, and I'll see everybody later.
You too. Thank you, Danny.
All right. Bye-bye.